Korea has overcome so many obstacles and achieved remarkable economic growth. And now it has transformed into a country with cutting-edge technology and vibrant culture. Without Busan, Korea as we know today would not exist. A lot of global events have been held in Korea. And then there is 2030. Busan has been at the forefront of Korea's economic development. Now it is ready to host the World Expo 2030 and join hands with the international community to present a blueprint for a better future for all mankind. With the ongoing global crises, the World Expo 2030 Busan will be a turning point for mankind. An opportunity to discuss the shared vision for emerging technologies and sustainable development. A festival that people from all around the world can enjoy anytime and anywhere through digital platforms. Through global solidarity and cooperation, we will play a bridging role between developing and developed countries in charting a new course for the prosperity of mankind. Busan is the city that envisions the global pivotal state initiative of Korea, upholding the values of freedom, peace, prosperity, and human rights. Let's work together to build a better future by sharing experience and knowledge, transforming our world, navigating toward a better future. World Expo 2030, Busan, Korea.
We study food. Because that is the people's health. We study drugs. Because that is the people's safety. We study cosmetics. Because that is the people's happiness. In order to ensure that your daily life is safe and happy, our love for you is realized through innovative science. Science of love for our people, the Ministry of Food and Drug Safety. The bigger our love, the stricter we become. As an agency that faithfully performs its duties given by the people, everything that our people use, from food and drugs to quasi-drugs and medical devices, to health-functional food, cosmetics, and sanitary products, reflects our thoroughness. In order to keep our food table safe from adulterated foods, germs and viruses, and hazardous chemical substances, we manage the safety of food ingredients according to systematic standards, conduct on-site inspections of manufacturers, and thoroughly manage the customs process for imported food to block harmful elements in the first place. Through the hazardous food sales prevention system, we also respond immediately to safety accidents after distribution. As the country's food and drug safety control tower, we make South Korea a nation that puts people's safety first. The more we care, the more we listen. As we are well aware that our efforts have no meaning without the people's trust, we transparently disclose the safety management information of food and drugs to the public. We also make efforts to improve school meals for our children, take the lead in educating people on proper eating habits, and establish a social safety net based on communication through adverse drug reaction relief system. In addition, we operate the Korea Orphan and Essential Drug Center to swiftly provide crucial medicines to people and prevent drug abuse through systematic management of medical narcotic drugs in an effort to create a reliable society and a safe nation. our loving mind is growing into love for the world. We foresee the trends of advanced science and technology and implement food and drug policy on par with the development level of South Korea. With smart approval evaluation and an advanced management system at international levels, we drive the innovative growth of the drug and medical device industries. In addition, we provide technical support for all cycles of the bioindustry, which is at the heart of the Fourth Industrial Revolution. From clinical trials to approval for sale, as well as present international standards through scientific R&D to contribute to technological development for food and drug safety. Our relentless efforts are making South Korea one of the world's 5 powerhouses in the food and drug field. Relief beyond safety. Trust beyond relief. Happiness to the people. Vitality to the industries. We will increase the value of our country. We will prove our love for you by realizing the happiness of the people. Science of Love for Our People. The Ministry of Food and Drug Safety.
Korea has overcome so many obstacles and achieved remarkable economic growth. And now it has transformed into a country with cutting-edge technology and vibrant culture. Without Busan, Korea as we know today would not exist. A lot of global events have been held in Korea. And then there is 2030. Busan has been at the forefront of Korea's economic development. Now it is ready to host the World Expo 2030 and join hands with the international community to present a blueprint for a better future for all mankind. With the ongoing global crises, the World Expo 2030 Busan will be a turning point for mankind. An opportunity to discuss the shared vision for emerging technologies and sustainable development. A festival that people from all around the world can enjoy anytime and anywhere through digital platforms. Through global solidarity and cooperation, we will play a bridging role between developing and developed countries in charting a new course for the prosperity of mankind. Busan is the city that envisions the global pivotal state initiative of Korea, upholding the values of freedom, peace, prosperity, and human rights. Let's work together to build a better future by sharing experience and knowledge, transforming our world, navigating toward a better future. World Expo 2030, Busan, Korea.
We study food. Because that is the people's health. We study drugs. Because that is the people's safety. We study cosmetics. Because that is the people's happiness. In order to ensure that your daily life is safe and happy, our love for you is realized through innovative science. Science of love for our people, the Ministry of Food and Drug Safety. The bigger our love, the stricter we become. As an agency that faithfully performs its duties given by the people, everything that our people use, from food and drugs to quasi-drugs and medical devices, to health-functional food, cosmetics, and sanitary products, reflects our thoroughness. In order to keep our food table safe from adulterated foods, germs and viruses, and hazardous chemical substances, we manage the safety of food ingredients according to systematic standards, conduct on-site inspections of manufacturers, and thoroughly manage the customs process for imported food to block harmful elements in the first place. Through the hazardous food sales prevention system, we also respond immediately to safety accidents after distribution. As the country's food and drug safety control tower, we make South Korea a nation that puts people's safety first. The more we care, the more we listen. As we are well aware that our efforts have no meaning without the people's trust, we transparently disclose the safety management information of food and drugs to the public. We also make efforts to improve school meals for our children, take the lead in educating people on proper eating habits, and establish a social safety net based on communication through adverse drug reaction relief system. In addition, we operate the Korea Orphan and Essential Drug Center to swiftly provide crucial medicines to people and prevent drug abuse through systematic management of medical narcotic drugs in an effort to create a reliable society and a safe nation. Our loving mind is growing into love for the world. We foresee the trends of advanced science and technology and implement food and drug policy on par with the development level of South Korea. With smart approval evaluation and an advanced management system at international levels, we drive the innovative growth of the drug and medical device industries. In addition, we provide technical support for all cycles of the bio industry, which is at the heart of the Fourth Industrial Revolution. From clinical trials to approval for sale, as well as present international standards through scientific R&D to contribute to technological development for food and drug safety. Our relentless efforts are making South Korea one of the world's 5 powerhouses in the food and drug field. Relief beyond safety. Trust beyond relief. Happiness to the people. Vitality to the industries. We will increase the value of our country. We will prove our love for you by realizing the happiness of the people. Science of Love for Our People. The Ministry of Food and Drug Safety.
Korea has overcome so many obstacles and achieved remarkable economic growth. And now it has transformed into a country with cutting-edge technology and vibrant culture. Without Busan, Korea as we know today would not exist. A lot of global events have been held in Korea. And then there is 2030. Busan has been at the forefront of Korea's economic development. Now it is ready to host the World Expo 2030 and join hands with the international community to present a blueprint for a better future for all mankind. With the ongoing global crises, the World Expo 2030 Busan will be a turning point for mankind. An opportunity to discuss the shared vision for emerging technologies and sustainable development. A festival that people from all around the world can enjoy anytime and anywhere through digital platforms. Through global solidarity and cooperation, we will play a bridging role between developing and developed countries in charting a new course for the prosperity of mankind. Busan is the city that envisions the global pivotal state initiative of Korea, upholding the values of freedom, peace, prosperity, and human rights. Let's work together to build a better future by sharing experience and knowledge, transforming our world, navigating toward a better future. World Expo 2030, Busan, Korea.
We study food. Because that is the people's health. We study drugs. Because that is the people's safety. We study cosmetics. Because that is the people's happiness. In order to ensure that your daily life is safe and happy, our love for you is realized through innovative science. Science of love for our people. The Ministry of Food and Drug Safety. The bigger our love, the stricter we become.
안내 말씀드립니다.
As an agency that faithfully
네, 본 컴
Ladies and gentlemen, we are holding a hybrid conference, and we will begin today's program from 1 o'clock. I would like to ask participants here at the venue to please take your seats. Simultaneous interpretation is provided throughout the conference. If you need translation, please remember to pick up a translation receiver from the information desk. Also, please be advised that we have provided you with proceedings and a feedback sheet. Please remember to fill in the feedback sheet and submit to the information desk so that we can prepare a better conference for you in the future. Those of you joining us online, please use the chat window to enter your comments, capture that, and submit that to the conference official homepage. And some of you will be receiving gifts from us. The conference will begin shortly from 1. Please take your seats. Thank you.
We disclose the safety management information of food and drugs to the public. We also make efforts to improve school meals for our children, take the lead in educating people on proper eating habits, and establish a social safety net based on communication through adverse drug reaction relief system. In addition, we operate the Korea Orphan and Essential Drug Center to swiftly provide crucial medicines to people and prevent drug abuse through systematic management of medical narcotic drugs in an effort to create a reliable society and a safe nation. Our loving mind is growing into love for the world. We foresee the trends of advanced science and technology and implement food and drug policy on par with the development level of South Korea. With smart approval evaluation and an advanced management system at international levels, we drive the innovative growth of the drug and medical device industries. In addition, we provide technical support for all cycles of the bioindustry, which is at the heart of the Fourth Industrial Revolution. From clinical trials to approval for sale, as well as present international standards through scientific R&D to contribute to technological development for food and drug safety. Our relentless efforts are making South Korea one of the world's 5 powerhouses in the food and drug field. Relief beyond safety. Trust beyond relief. Happiness to the people. Vitality to the industries. We will increase the value of our country we will prove our love for you by realizing the happiness of the people. Science of Love for Our People, the Ministry of Food and Drug Safety.
안녕하십니까?
Good afternoon. This is the second day of the third GCFA. My name is Chung Ji-yeon from the Livestock Product Safety Policy Division of the MFDS. I will be leading you through today's program. It is raining outside. In spite of that, we have many people here in spite their very busy schedules. So I would like to thank all of you from both home and abroad. Also, many people are joining online as well, so thank you very much for your participation. So, like we did yesterday, we are broadcasting today's conference live on YouTube, like we did yesterday. And also, the video will be uploaded on the official conference homepage after the conference is concluded. So we had the wonderful keynote speech by Professor Park Yong-ho yesterday, and we had two sessions. The first session was efforts by international organizations to reduce AMR in the One Health approach, and we heard presentations from FAO, WHO, and OHA. And in session 2, under the theme of current status and strategies of AMR reduction in food, human, and environment, we heard from EFSA of EU, from MFDS of Korea. We heard very impressive presentations. And today, we will be having sessions on latest technologies for reduction and surveillance of AMR, and also the FAO Act project. And also we will have summary and discussion at the end. I really look forward to the content that will be shared today. So we will now begin the second day of the third GCFA, and this is session 3. Which is on the latest technology of reduction and surveillance of AMR. And let me introduce the chairperson of session 3. With the big round of applause, please welcome Kim Sun-han from the MFDS. Good morning. I am Director of Food Microbiology Division at NIFDS. And in this session, we will be learning the latest technologies for reduction and surveillance of AMR. There will be 3 presentations.
So the first presenter will be talking about the application of the whole genome sequencing for AMR monitoring program. And for this, Dr. Xiaohua Zhao from the US FDA will give us a presentation. He's a senior biomedical research and biomedical product assessment service at FDA Centers for Veterinary Medicine. She received her PhD from the University of California. And please welcome her with a big hand.
Good afternoon. First of all, I would like to thank the GCFA committee for inviting me to give this presentation. Today, I'm going to talk about the application of whole genome sequence for AMR surveillance program. Antimicrobial resistance poses significant threats to public health worldwide. Here is the US CDC 2019 AMR report. Each year, more than 2.8 million people infected with antimicrobial resistant bacteria in the United States. More than 35 people die as a result. Antimicrobial resistant Campylobacter and non-typhoid Salmonella, two major foodborne pathogens, were listed as a serious threat organism. To combat antimicrobial resistance, the US government, in partnership with foreign government and organization, has developed the US National Action Plan for combating antimicrobial resistance. The plan takes a One Health approach and set up 5 strategic goals. One of the goals is to strengthen national One Health surveillance efforts to combat resistance. Another goal is to advance development and use of rapid and innovative diagnostic tests for identification and characterization of resistant bacteria. The US National Antimicrobial Resistance Monitoring Program, NAMS, is working towards this goal. NAMS program was established in the 1996. It's a collaborative effort between FDA, CDC, and USDA. And the 3 agencies working together to determine antimicrobial resistant profile of Campylobacter and Salmonella isolated from human, retail meat, and food animals. CDC is in charge to test all the human isolates, and FDA in charge to test the bacteria isolated from retail meats. And all the food animal isolates were tested by USDA. In addition to Campylobacter and Salmonella, FDA and CDC also tested the antimicrobial resistant profile of E. coli and endococcites. CDC tested additional organisms such as E. coli O157H7, Salmonella typhi, Shigella, and Vibrio. For FDA, since we started sampling the seafood, so we tested additional 2 organisms, Aromonas and Vibrio. Each year, we have integration final report and posted our findings in the website. In order to improve the One Health surveillance program, in the last few years, we have collaboration with EPA, Environmental Protection Agency, to determine the resistant profile of bacteria isolated from surface water. We also collaborated with the veterinary laboratory investigation and the response network to determine the resistant profile of bacteria isolated from a companion animal. In the past, NAMS used several methods to characterize the bacteria isolated from a different source. We use traditional biochemical and the PCR for bacteria speciation. We also use broth microdilution to determine the resistant profile. Use traditional serology to determine Salmonella cell type. We also perform post-field gel electrophoresis, PFG, to determine bacteria clonality. When we report the trend of the resistance, very often we do not know what gene or mutation are corresponding of this phenotype. So we will take additional research to find out the resistant mechanism. We normally use the PCR followed by sequence analysis to identify the gene or mutation that are responsible for those phenotype. As you can see, this is a very tedious process and very costly. In the last 10-15 years, whole genome sequence has improved tremendously become cheaper and faster. So we are thinking that use the whole genome sequence, maybe we can replace the multiple methods I just mentioned in my previous slide to characterize the bacteria. And before we implement the WGS to the NAMS program, we want to make sure that we'll be able to detect all the resistant gene or mutation from the WGS data. And also make sure that the resistant gene are responsible for the phenotype we tested. Here is an example. This is E. coli from phenotypic testing that show resistant to 5 different antimicrobial, resistant to streptomycin, tetracycline, chlorphenicol, sulfamexazole, nalidic acid. Through the whole genome sequence, we'll identify all the gene and mutations that are responsible for each of this phenotype. So in this case, we say the phenotypic genotypic correlate 100%. So before we implemented the WGS to the NAMS program, we have conducted several pilot projects to check the correlation between phenotype and genotype for the 4 organisms. Here is a Salmonella study. In this study, we selected the 640 Salmonella that represent 43 different serotypes isolated from retail meats and human. Those Salmonella has a different resistant profile and a different PFG pattern, isolated from 2011 and 2013. Through the whole genome sequence, we are able to identify 65 unique resistant genes, and plus the mutation in tooth housekeeping gene that are associated with quinolone resistance. The 65 unique genes that are responsible, they resistant to different antimicrobial classes and different antimicrobials I listed here. This slide shows the summary of the correlation between phenotypic and genotypic among the 640 Salmonella. On the left side here lists the 14 antimicrobial we tested for the phenotype. So we did the correlation, use the term sensitivity and specificity to see the correlation. Now I'd like to explain how we calculated the sensitivity and specificity. I use genomycin as an example. Among the 640 Salmonella isolates, 105 of them show the phenotypically resistant to genomycin. So 99 of them have carried the genomycin resistant gene. 6 of them do not carry the genomycin resistant gene. So 99 divided by 105, that's equal 94.3%. That is the sensitivity. Also, we call the positive correlation. Now let's look at the specificity. Among the 640 isolates, 535 show the phenotypically susceptible to genomycin. 530 did not carry any genomycin resistant gene. However, we have 5 isolates that carry the genomycin resistant gene. So 530 divided by 535, that's equal 99.1%. That is the specificity. So we did this calculation for each antimicrobial. As you can see, most of the isolates has over 100. Some of them are to 100% sensitivity and specificity. So overall, we got the 98.8% sensitivity and 99.1% specificity, which is greater results than what originally we expected. And you can see some of reach 100% sensitivity and specificity. However, some of them has lower, specificity, such as streptomycin, and some of lower sensitivity, such as trimethoprim. We only have the 86.4% sensitivity. So we did the further study to wonder why some of the correlation has a lower sensitivity and specificity. So we used the streptomycin as an example. So since CSI did not establish the resistant breakpoint for streptomycin for Salmonella and E. coli, so in the past, our program has used 64 microgram per ml as a resistant breaking point for streptomycin. So in this study, we used over 600 Salmonella and E. coli. So we run the phenotypic test to show how the MIC distribution look like for the isolates that with or without the streptomycin resistant gene. So as you can see, if isolates do not carry any resistant gene, then MIC is between 2 to 16. If isolates carry the streptomycin resistant gene, the MIC will be between 16 or over greater than 64. So if we use the 64 as a resistant breaking points to do the correlation, then we will miss some of the isolates with MIC 16 or 32, but they carry the streptomycin resistant gene. So now let's look at the right side of the table. As you can see, if we use 64 as break points, we maintain the sensitivity 100%, but the specificity only 68%. If we lower down the break points to 32, we still get the 100% sensitivity, but the specificity increased to 92.6%. If we use the break points to 16, now the specificity improved to 100%, but the sensitivity decreased to 97.9%. So based on this study, our program has decided to use 32 microgram per ml as a resistant break points. Streptomycin study, so we think that we can establish the user WGS to determine or establish the genomic cutoff value to measure the antimicrobial resistance. So in this study, we used over 1,700 Salmonella isolates to investigate the isolates with the gene, without the antimicrobial resistant gene, how the MIC distribution look like. So here we use ciprofloxacin here as an example. So any isolates that do not carry the quinone resistant gene, that they have the MIC equal or less than 0.06. This is over 1,600 isolates, but there's except one. And the rest of the isolates, they carry different quinone resistant mechanism. They have MIC equal or greater than 1012 microgram per ml. So depending on what mechanism they have, they don't have different the MIC. For example, for these three isolates, they carry the double mutation in GIA. They have MIC greater than 4 microgram per ml. So we finally to look at the comparison of genomic cutoff versus the CLSI and UCAS break points. Here we analyzed the 13 different antimicrobials. Here is the CLSI susceptible break points. In other words, any isolates have the equal or less this value. That means they're susceptible to each of this drug. Now let's compare the GCFA. With those values equal or less this value, that means those isolates do not carry the resistant gene for those drugs. So as you can see, compare GCFA and the CLSI break points, they have great correlation. Many of the value identical. Some of our GCFA value also are correlated very well with UCAS, the break value. This slide shows the pilot project to look at the phenotypic genotypic correlation on Campylobacter. In this study, we used 114 Campylobacter isolated from retail meat, human, and the food animal. And we determined the phenotype with the 9 different antimicrobial belong to 7 different classes. So through the whole genome sequence, we are able to identify the resistant gene that are responsible for each of this phenotype. Then we calculate the sensitivity and specificity just like we did on Salmonella. As you can see, many of the isolates, show the great correlation with the phenotype. And some of drugs showed 100% sensitivity and specificity. For example, tetracycline and quinolone, these two drugs showed 100% sensitivity and specificity. So overall, we got the 99.2% sensitivity and specificity. So this results are even better than Salmonella data. So we continue conduct the pilot project for enterococci and E. coli. All of the data show the great phenotypic genotypic correlation. We published all this data in peer-reviewed journal. So if you are interested in detail of our finding, and you can read our publication I listed here. So since 2015, we have implemented WGS in the NAMS retail meat program. We sequence every single Salmonella and Campylobacter isolated from retail meats in 2015. And in 2019, we started sequence all the E. coli and some of enterococci. And in 2019, we transition sequence to NAMS retail meat sites. The WGS data generated from sites is directly submitted to NCBI. And this allows us in the real time to share NAMS resistant data to public, and allow us more rapid for outbreak detection and response. Also, we learned, gained the knowledge of the emerging resistance. Now, I like to give the few examples how we use WGS to support AMR research. First example is detecting novel gentamicin resistant gene in Campylobacter isolated from human, retail chicken, and food animals. The prevalence of gentamicin resistant Campylobacter in the past is quite low. However, in the 2011, there's a sharply increase of gentamicin-resistant Campylobacter coli from human and retail chicken. So the initially objective of this study, we wonder what is the resistance mechanism, which genes are responsible for this, sharp increase of gentamicin resistance. We run the PFG, and with the two enzymes, all the chicken isolates and human isolates show the identical PFG pattern with two enzymes, and also same resistant profile. They're all co-resistant to tetracycline except one isolate. So this data analysis show that the human infection is due to the consumption of contaminated chicken. Based on the literature, we have designed the primer to detect the gentamicin resistant gene that is called APH family. But the PCR results was have poorly reproducibility. We couldn't get a consistent results. So finally, we decided to do the whole genome sequence. Through the whole genome sequence, we identified the 9 gentamicin resistant genes, and 7 of them are monofunctional, and 2 of them are bifunctional. 7 of them I highlighted red here. Those gentamicin resistant genes have never reported in Campylobacter. And APH2IG, I highlighted in yellow here, that is gene are responsible for 2011 outbreak. The APH family is quite diverse. So you can see here, amino acid identity can be as low as 26%. So this is explain why our PCR didn't work. We had a very poor reproducibility. We have closed the genome and the plasmids for several, this gentamicin resistant Campylobacter coli. They all carry the PTAT MDR plasmids. So they carry the APH2IG gene, but also they carry additional amyloglycan resistant gene as well as the tetracycline TIO gene. So this explains why they all co-resistant to tetracycline. The seminar plasmid carry the seminar resistant gene has been found in Campylobacter isolated from chicken in China. Now I like to give the second example of our study, emerging of ESBL 3665 produced Salmonella infantis clone from human and chicken in the United States. Salmonella infantis is a top 6 serotype that causes the human infection in the United States. The ranking has increased over last decades from top 12 to top 6. Here is the CDC's report. Compare the 2016-2018 to 2019, the top serotype of the incidence, as you can see, except the Salmonella infantis, all the top serotype has decreased in the last few years. However, Salmonella infantis has increased 69% the incidence. This is really significant. Salmonella infantis also has increased the prevalence from the retail chicken. And in the 2014, for the first time, we have identified the Salmonella infantis that carry CTX65. And also the increase we see in the human isolates and the chicken seca sample. So we sequenced the several of this Salmonella infantis isolated from a chicken seca sample, retail chicken, and human. All of those isolates carried the Mego MDR plasmids. These plasmids, besides carry the CTX65, they carry many aminoglycosides resistant gene, trimethylprid resistant gene, fluorophenical resistant gene, phosphor A gene, also tetracycline resistant gene, sulforamide resistant gene, and the streptomycin resistant gene. In addition, this clone has the GIA mutation, so phenotypically they also show decreased susceptibility to ciprofloxacin. This clone has widely spread in the US. As I mentioned here, in the 2014, we first detected this clone in Tennessee. By the 2019, every NOMS retail meat size has detected this clone. Let's look at the global level of this clone. It was first reported in South America, Peru in 2010. By the 2020, this particular clone has reported in many countries. In South America, North America, Europe, Russia, and Asia. So this is a great concern. This particular clone significantly increased from 2010 to 2020 in the retail chicken in the US. But we also found this clone in ground turkey and pork chop. And this CTX65 plasmids also spread to other serotypes. And this plasmids is conjugative, spread to the different strain. So this is a great concern because this particular clone showed resistance to 8 or 10 different antimicrobial and decreased susceptibility to ciprofloxacin. And so because ceftriaxone and the ciprofloxacin are drug of choice to treat the Salmonella infection. So those clones show the resistance to these two drugs. Now, I'd like to give an example. We use the long-read sequence technology, such as a backpile, to close the genome and to study the genomic structure of multi-drug resistant plasmids and the virulence of foodborne pathogens. Here, just give the example, the two Salmonella serotype, we have closed the plasmid and genome. The first isolates here is Salmonella typhimurium, isolated from swine. These isolates carried the 3 plasmids. The big plasmids has over 200 kilobase pair, and they carried the 8 antimicrobial resistant gene highlighted in the red, and also carried the 8 mercury resistant gene, heavy metal resistant gene. This plasmid is a hybrid plasmid. The second plasmid is 94 kilobase plasmid, is also hybrid plasmids carry the marker for Incan F2 and Incan FIB. This plasmid is a valence plasmid because they carry the valence operon SPV RABCD. The valence operon has highly associated with the severity of Salmonella infection. The smallest plasmid here is untypable, but it carries the 6 antimicrobial resistant gene highlighted in the red, and 1 disinfect gene, and also 7 of the mercury resistant gene. In addition to the plasmids, this particular isolate carried the Salmonella genomic isolate in chromosome. So in this genomic isolate also contained 6 antimicrobial resistant gene and 2 disinfected gene. So this particular isolate totally carried the 20 antimicrobial resistant gene, 17 heavy metal gene, and 3 disinfected resistant gene. So you can imagine how difficult to treat such infections. Now let's look at another isolate. Salmonella doubling. This is a plasmid structure, and this is a chromosome, but it's the same strain of the Salmonella doubling. This is a plasmid over 300 kilobase pair. Is a hybrid MDR valence plasmids because valence plasmids has integrated to MDR plasmids. They carry the valence operon SPV RABCD. And they also carried the 12 different antimicrobial resistant gene. This plasmids, if you do the traditional typing, you may think that this isolates carry 5 different plasmids. But when we close the genome, we showed that there's a single plasmids. Actually, they carry the 5 different marker of the plasmids here. So look at the chromosome here. The chromosome show that this Salmonella doubling carry the 15 different Salmonella pathogenic isolate. They evenly distributed to the chromosome. Salmonella, the Salmonella pathogenic isolate involved the serotype evolution and also the pathogenesis of the infection. Now, finally, I'd like to just briefly mention about the application of beta genomic for resistant profiling. This study was led by my colleague, Dr. Daniel Tadesse. This is a One Health approach. We like to use metagenomic to look at the resistant profile from different commodity. The red circle here showed the resistant profile from the surface water. And the blue circle here shows the resistance from the different food animal, including swine, cattle, turkey, and chicken. If you compare surface water and the food animal, they do share, many of them are resistant gene. And also, if you compare the food animal, they also share the resistant gene here as well too. However, each commodity, they have their unique resistant profile. This is very important. I think in the future, the metagenomic probably can give us a much more comprehensive resistant profile from each commodity. This can really strengthen our AMR surveillance program. So in summary, I hope our data has shown you the power of the whole genome sequence for AMR surveillance and research. And the WGS has revolutionized our AMR surveillance program, and our program has been truly transformed from a phenotypic AMR surveillance to genotypic AMR surveillance program. And WGS can serve as a single assay for the program and supplement multiple methods, save time and money. Finally, I'd like to acknowledge my colleague from FDA Center for Veterinary Medicine. They have been working so hard in the last 20 years to continue improve the NAMS program. And also I'd like to thank my colleague from other agencies, CDC, USDA, NCBI, and the state public health laboratory and some university, they are funded by NAMS retail bids. They make the significant contribution and for the program and make the NAMS become an excellent AMR surveillance program. Thanks for the listening.
네, 첫 번째 발표.
Yes, that was the first presentation, but this was a recorded tape, and so we will not have a separate Q&A session, and we will go right into the second presentation, which will be on combating AMR, an overview of bacteriophage therapy, and Professor Yu Sang-yeol of SNU will be giving this presentation. Professor Yu received his degree, bachelor and master's at Seoul National University, and also he received his PhD in food microbiology from the University of Massachusetts. Let's welcome him with a big hand.
예, 감사합니다.
Thank you very much. So today, during this talk, or during this session, we're trying to utilize different methods in order to deal with the issue of AMR. So there's a lot of recent technology. And recently, there's a lot of interest in bacteriophage therapy, and that is what my talk is going to be on. As you all know, at a global level, we have different antibiotics, and we are seeing there are organisms that have developed resistance. And there is more concern, bigger concern now, because we have been using more antibiotics because of the COVID-19 pandemic. So this means that there will be more organisms in the future resistant to these antimicrobial agents. So one solution could be bacteriophage. So this is a type of virus. However, it infects only bacteria, and it is harmless to human cells and also animal and plant cells. And in the world, on the globe, there are 10 to the 31st power to 32nd power macrophages in the world. So it's very, very abundant. And by these bacteriophages, 50% of the bacteria that exist die in a day, and then they regenerate. And there are two cycles in the bacteriophage life cycle. And so the bacteriophage goes into the host, it kills the host, and then it comes out. And then also it can go inside the chromosome of the host, and it will be part of the chromosome. So that's part of the life cycle as well. For therapy use, we will be using the lytic cycle that you see on the left side of the diagram. So the lytic cycle. And if you look at the genome, you can see that it has great diversity, ranging from 17 kilobase pair to 500 kilobase pair. And bacteriophages are very, very numerous and high in number. And so even if we analyze the smallest gene, there are some genes that we find where we cannot find out the function, and that's about 15% of the total genes. And so this means that this can provide new genes, novel genes for numerous applications. So this is history. So we found out about phages in 1890s to 1900. So there was this person called Hankin, and he observed antibacterial activity against Vibrio in the Indian river water. And after that, it was confirmed that bacteriophage is effective in dealing with bacteria. And before we had antibiotics, we were using bacteriophage to treat certain diseases, certain infections, and it was used as a panacea, and it was commonly sold in drugstores and pharmacies. However, there was a problem because at that time people did not know that bacteriophage had specificity for bacteria. So sometimes it did wonders for the disease, sometimes it did nothing. And then since the 1940s, antibiotics were developed, and in the West, antibiotics were used. So bacteriophage use decreased drastically. However, in Soviet Georgia or in the East, there was continued research. And you can see that we have a research institute that has been established, and they celebrated their 100th year this year. However, now we have the problem of antibiotic resistance and now renewed interest in bacteriophages. And in year 2006, US FDA approved the use of phage cocktail for the use in RTE meats to prevent listeria. Now, there are two characteristics of phage. The first one is that it shows highly specific infection. And we have metagenome, where there's a lot of interest here. And there will be no issues raised in the microbiome, and it can target the specific bacteria. So that's one big advantage of bacteriophage. Another thing is that when it goes inside the host, it amplifies tenfold to 1,000-fold. We can use these characteristics, and we can detect a very small number of pathogens by using macrophages because they amplify so fast. And so how can we use these characteristics for therapeutic use? So that is where we will have to do future research. So we have the phage, but we have other things that we have to consider as well. We have to think about lysis, which has to happen. And lysis, well, we have the lysine, which is the enzyme that is created by the phage, and this is degraded by glycan, and we can use the phage for therapeutic uses. But not only that, we have endolysin, which is a byproduct of this process, and we can clone this outside the cell, and we can use this material in order to break down cell walls, and that can be used for therapeutic uses as well. So I would like to talk about phage today, and also endolysin, how we can utilize these two different materials. So in this table, I compare antibiotics and the bacteriophages and the endolysin. So basically, for antibiotics, specificity is very, very low. And so if you give antibiotics, most of the bacteria will die. However, there is specificity for the phages and the endolysin, and so we can minimize side effects. Also, the system is quite simple. It's very easy to develop. These are some major characteristics. Moreover, we may have resistance against bacteriophages as well. However, the bacteria that develops the resistance has very little pathogenic characteristics. And so receptors are used to enter cells, and these receptors play a role so that the bacteria that has resistance against the bacteriophage has lost part of its component, and it is no longer so pathogenic. And so when we use phage, there could be some resistant bacteria. However, it's less pathogenic, so it may not be a big issue. So that's a very big advantage. And so as you can see here, many companies are working on the development of bacteriophage in order to develop drugs and treatments. So first of all, let me explain phage therapy. So as I said, there are diverse phages, so if we play the game correctly, then we can target the exact bacteria that we do want to target. And so here I want to talk about the Salmonella phages that we are studying at our lab. So we have isolated dozens of such phages, and there are short ones, long phages that is. And then there are ones with tails that contract. So there are three major categories. And so in our case, the bacteriophage will go inside the host. So we did a lot of study on the receptors. And the host range is quite narrow, so we mix the phages, so we call them phage cocktails. And usually we mix 3 to 4 different types of phages, and we try to control specific strains of bacteria. Now, in order to mix the different types of phages, maybe we can utilize phages that have different receptors. And when we have resistant bacteria in the future, we can lower the possibility of that happening by having these different receptor types in one single cocktail. So we have TN5 random mutant library. We have utilized that. And we screened the receptors of the phages that we found. And the result is what you see here on this table. So we looked at the receptors of 20 plus phages. And as you can see here, it's quite interesting because they roughly fall into 3. So it's either Flagella or BTUB or LPS. These were the 3 different receptors that were being used. So 3 groups existed. And so in each group, we did a bacterial challenge assay. We wanted to pick the one that did the best job in killing the bacteria. And by using the 3, combining the 3, we came up with a cocktail. And the morphology of the selected phages are as you see on the screen. Now, this is the genome sequencing that we did for the 3 different phages. And we looked for specific pathogenic genes, organomes, or we made sure that they did not have the genomes that we did not want, and that they are indeed the right candidates for the cocktail that we're trying to make. And so we selected the 3 bacteriophages, and you can see the host range analysis that we did for the 3 phages. And we test for Salmonella, and you can see that all 3 were effective. And as for BSP-101 phage, it's quite unique because it works for Salmonella, but also E. coli and also O157 pathogen. It was quite effective in controlling all 3 of these pathogens. Now, as for BSP-101, we have the O antigen. So it uses the receptor that the BSP-101 uses is the O antigen. So judging from that, maybe it has to do with the O antigen structure of the Salmonella. It's quite similar, and that's why it's effective. So we chose the 3 phages, and we did the bacterial challenge to check out the effectiveness. And here you can see the efficiency of the 3 phage cocktail. And so you can see the control and the individual phages working, and then you can see the efficiency of the cocktail. We also looked at how often mutants are likely to occur. So in the 3 phage cocktail, you can see that compared to mixtures of 2 or just single phages, you can see that the BIM is actually lower. And also we took iceberg lettuce and cucumber and did actual exams. And you can see that there is a reduction effect of more than 5 logs. And we isolated 2 other phages and we mixed them together. And we did some mice experiments. And usually the mice, well, we have salmonella infection and usually they die within 10 days. However, we had MI10, so multiplicity of infection of 10 is the infection that was given. And so that is the ratio between the bacteria and the bacteriophage. And we used the infection factor of 10, and only with that you can see that it was quite effective against salmonella, and the mice was perfectly protected from salmonella infection and it was able to live. And we can think of applying this to food and also mice, and you can see that in the past 2 experiments our cocktail is very effective. So as you have seen thus far, there could be numerous applications for bacteriophages. And during what I talked about, well, I talked about narrow range of host specificity. This could be an advantage. In some cases, if we cannot find the right host range, then we cannot use the phage for therapeutic effects. So perhaps we can manipulate and we can do phage engineering to make that match. And maybe we can extend the host range of phages. So we did an experiment on that and I would like to share the results. So generally, we have the tail spike protein in the tails of phages, and they actually determine the host range. And so we looked at the numerous phages that we had, and we tried to substitute the different tail spikes in the receptors, and we looked at how that changed the host range. And so this is the tail spike protein of the phages that we have, and we have these results, and then we also have other published information from other labs. And amino acid sequence residue that was in the range of 400 to 450, and they played a very important role in interacting with other proteins. And so based on that information, this is what we did. So we have P22, TS3, T56, 32H, and so forth. So we looked at the tail spike protein in these tails, and we used CRISPR-9 technology, and we came up with this hyperphage. And so the protein sequence from P22 was switched with the tail spike protein in the receptor of 32H and so forth. So we did switching of the tail spike protein, and we have compared the wild type and the mutant. And the marker that you see here on the bottom, you can see that there was structural change to the genes, and you can see that specificity changes following the switches that we made. And the LPS structure changes, and accordingly, the host range changed as well, and we could manipulate it in the direction that we wanted. And here, well, you can see that there are many different organisms that are resistant against antimicrobial agents. And so we work on choosing the right media, and we also are having difficulty in isolating our target bacteria. So Campylobacter jejuni, in that case, they have resistance. And so here we need selective media. So we have Bolton broth and Proston broth that we used. And when we put in different antibiotics, well, we can see other bacteria contaminating the broth, and we can actually work on these other contaminating bacteria in order to kill the Campylobacter jejuni. And so, even when we use antibiotics, we can see that many of the organisms in there do not die, and it is very difficult to isolate the target bacteria. So that is an issue that we are experiencing recently. And so here we're doing higher Campylobacter jejuni isolation in the presence of phages targeting E. coli. So we want phages that can kill the E. coli so that we can better isolate the Campylobacter. And we compare the efficiency in isolating the Campylobacter, and we tested it in 2 different temperature ranges. So the gray bars show the phage cocktail. So it's not 100%, but compared to the control where we did not have the phage cocktail, you can see that when we do use the cocktail, we have higher efficiency in isolating our target bacteria. And so if the sample is contaminated with these other resistant bacteria, well, then we can utilize phage cocktail to kill the non-target organism in order to isolate our target bacteria easily. And recently, well, of course, we have tried to find alternative to antibiotics, but that is not easy. And so we thought of maybe using phages and antibiotics together. And if that is more effective, perhaps we can use less antibiotics. So we have tetracycline, ampicillin, and so forth, so commonly used antibiotics. And then we combine that with phages and then try to see how effective that was. And we could not really see a clear trend there. However, BTUB, when phages use this as a receptor, when we use kanamycin at high dose, you can see that it was effective and the bacterial level did go down. And so perhaps we need to do more future research to think about how we can use phages and antibiotics together to have better effects. Next, I would like to briefly go over the topic of endolysin. So after the phages grow in number inside the cell, it tries to break out and they produce endolysin. And we can actually go through this process outside the cell wall, and it is effective, and it could be an alternative to antibiotics. So basic structure of endolysin. So we have 2 domains. They are linked together. One is the enzymatically active domain, and the other is the cell wall binding domain. As for the latter, it is more effective than antibodies, and it binds better to cells and cell walls. We can use this in order to come up with treatment. So it is a very unique enzyme. And so, for example, for S. aureus, we have lyse SA97 that works on that. This is a endolysin. And when we isolate and do the treatment, as you can see here, it is a concentration or dosage dependent result. And what is important here is that we have S. aureus pathogen. It is a serious issue because they form biofilm. And when we apply antibiotics, when we apply drugs, it cannot enter the cells. It cannot show effect because it cannot go through this biofilm. However, here, as you can see, lyse SA97 effectively disrupts the biofilm that is created by S. aureus. Also, we have carvacrol. This is a naturally occurring antibiotic, and we can combine this with Lyse SA97. And we looked at the synergistic effect that it brought. So we have, you can see the effect when they are used as single agents. However, when we use 1/10 of the two in amount, combine them, and then use it, they have the tenfold effect of using the ingredients as single agents. So if you look at SEM photos, you can see that the cells of S. aureus are effectively deformed by using this cocktail. And at this time, 100 S. aureus endolysins were studied, and the diversity was not really high. We could see that roughly they could be divided into 6 different categories. And so we have the cell wall binding domain and the enzymatically active domain. That was quite unique because in type 1 and 2, they only had one. And then if we look at the other endolysins in the enzymatic active zone, it exists as singles. However, from type 3 to type 6, we can see that there are 2 enzymatic active domains. So that was quite unique. And so we had an idea here. From the perspective of the bacteriophages, there could be endolysins that are very effective, and that is sent out from the bacteriophages, and that could kill off all the bacteria that it might actually need in the future. So in nature, I believe that the bacteriophages, they exclude endolysin, which has a lesser effect, so that they can survive in that environment in the future. And so in the lab, we did in vitro tests. And perhaps it is highly likely that we can enhance this less than 100% effectiveness. And so we have numerous domains of the endolysin, and we did random shuffling of them, and we switched the positions around. So in nature, compared to what is in the nature, we are trying to find endolysin that is more effective. So we used a 2-vector system to do the expression, and then we did some screening. So we have a system of doing that. And as you can see here, what you see that is kind of black is the size of the inhibition zone is quite high when it shows up in dark color. So we can use this, and we can do the domain shuffling randomly, and then we found the ones that are more effective. And one that we found to be effective is Cli-C, and the mother form of Cli-C would be Lyse SA12. And we used 2 concentrations here to do this test, and in both concentrations, Cli-C was more antibacterial in its effect compared to Lyse SA12. And some characteristics of Cis-C. So temperature, in very high temperatures, not much of a difference, but at about 45 degrees, when we use it for therapeutic purposes, we can use it at 45 degrees for humans or animals. It was quite stable at these temperatures. And then pH was these levels. However, NaCl, you can see that Cli-C at certain levels had less stability compared to Lyse SA12. And the biofilm disruption capability of Cli-C was higher. Also, we did tests in milk, blood, and serum. And in all 3 cases, you can see that Cli-C always was the more effective compound. And where we used Lyse SA, and we did some mice test, and you can see that we gave 10 milligram per kg of the mice. We used the glycine, and you can see that when we used it at that dose, it provided ample protection of mice. So we isolated different types of endolysins, and we have a certain bottleneck because the problem is, in order to isolate the endolysin, we have to isolate the bacteriophages first, and that can be sometimes easy, sometimes difficult. And so now we have a recent technology that has been developed, and we utilize the shotgun metagenome mining method. And we can use this to perhaps isolate endolysins that we were unable to isolate before. And from there, we can combine different DNAs and then come up with endolysin genes and genome that we were unable to isolate before. So we have these different types. Some work on Salmonella, some work on E. coli, some work on S. aureus, and so forth. So they show different characteristics. So by utilizing this technique, I think that we will be able to isolate more endolysins in the future. And lastly, I would like to talk about this. Endolysin that we have isolated before usually work for gram-positive germs. However, for the gram-negative, they have this membrane, and it is difficult for the endolysin to reach the gram-negative bacteria because of the outer membrane. Now, that is why we need some membrane destructing or destabilizing material. And so we're trying to use endolysin along with peptides that can destabilize the membranes. And of course, we have many genes within the bacteriophage where we do not know the characteristics. However, there are hydrophobic proteins, and there is some likeliness or some possibility that these proteins will be able to destabilize the membranes and enter the area of interest. So there are some possibilities that we see here. And here we did some fusions and we got some clones and we're carrying out experiments. Also, there is something called sensitizer peptides. So there are certain peptides that have that membrane disrupting capability, and then we can fuse that with endolysins for better effects. And you can see that the antibacterial activity of the fusion endolysin is quite effective. So here we're using insects in order to study against E. coli and also the Acinetobacter bacteria. And also we have something called holin or phage holin. And we saw some antimicrobial effect from that as well. And also the phage holin showed different effects. Some were very effective against these different types of bacteria. So I introduced you to different research on bacteriophage and endolysins, and perhaps in the future we can utilize this technology and study as a alternative and also supplementary method to dealing with the issue of AMR. So on that note, I would like to conclude my talk. So acknowledgements, students from our lab, they're working very hard, so I want to thank my students. Thank you.
네, 감사드립니다.
Thank you very much, Professor. Well, time is limited, so I think we have time for maybe 2 questions. Any questions or comments from the floor? Yes, please.
Thank you very much for a really interesting presentation and an extensive one. I'm curious to know, in the case of Korea, how many of these products, if any, are licensed already for use in either in food for decontamination or as medicinal products to treat disease in humans or animals? Do you have already any examples? And if that is not the case, could you tell us your views about potentially challenges in the regulation of such products and what can be making difficult to bring to the market these solutions? Thank you.
Nothing is licensed in Korea yet. So one phage product is licensed to be used as a feed additive, but nothing more. And I think Korean FDA should be more brave to give a license to this kind of products. But Korean FDA is too conservative, so it's not easy to get a license. Okay. So,
thank you for that great presentation, Jeff Lejeune from FAO. First, I'll disclose my bias. I am a phage proponent. I like the idea. We've got some work to do. But people who are against it argue two main things. One, that bacteria will mutate and then the phages won't work. Okay, and I don't have a problem with that because phages replicate a whole lot faster than bacteria and they mutate a whole lot faster than bacteria. So it's a cat and mouse game and the phages can catch up with the bacteria. The other one though, and I struggle with this one, is how do we make sure that the phages don't disseminate antimicrobial resistance genes? Because they can pick up genes and transfer them, and we could be acting counterproductively, trying to kill the bacteria, but instead enhancing them.
Okay, for your first question, bacteria can be resistant to any phage, but as I told you, the resistant bacteria normally lose its virulence. So the bacteria resistant to the bacteriophage normally cannot cause any disease. And we make phage cocktails to reduce the probability of resistant mutation. So that if you mix 4 or 5 phages together, you can reduce the chance of resistant development very efficiently. And for your second question, bacteriophages have 2 life cycles. So for the gene dissemination, the lysogenic cycle is very important. So let me show you. Can I see my slide again? Or? Okay, this is the 2 kinds of bacteriophage life cycle. So when the bacteriophage use this lysogenic cycle, that the certain genes can be transferred to the other bacteria, called the vertical transfer. So for the bacteriophage therapy, we use bacteriophage that do not have this lysogenic cycle. That phage have only this lytic cycle. So that vertical gene transfer probability is quite low. It's not 0%, but it's quite low. So you don't have to worry about dissemination of the resistance gene or virulence gene when you use this lytic cycle phages. Okay?
교수님께서
Now, Professor Yu you have said that the Korean MFDS should be more brave. Well, I'm not in charge of approving such products, but I will do my best. Maybe we can provide you with a team of product support. And yes, we are becoming braver and courageous. So yes, so thank you very much for that comment. And on that note, we would like to wrap this presentation up.
CNS의
Now moving on, we will have the last presentation of session 3. So the presenter is Mr. Rune Hendriksen, professor of the Technical University of Denmark. And he will be talking about new technology for AMR surveillance. Professor is currently working at the Technical University of Denmark, and also he is an external advisor in the Europe CDC, and he has served as a various advisory roles. So please welcome him with a big hand.
Yeah, so thank you for inviting me. It's a great pleasure to be here and talk about some of the topics that I like most, which is AMR and whole genome sequencing. So basically, my talk is more about the perspectives in the direction that I believe that we are moving. And I think that I would start with a quote, as also Professor Park started with, from Maggie Chan. And I'm not talking about how dangerous AMR is. I think we all know that. But I think that it's important to stress her last line about the need of a harmonized and standardized approach for surveillance in a One Health context. I think this is really the goal that we are trying to achieve. So we already have a lot of surveillance systems in place. These are built on phenotypic data. We have, as also Park mentioned, national surveillance, we have regional surveillance, and we have global surveillance. But again, when we look more into the data, the question is how reliable they are. So again, we have the surveillance in order to take action. But if we take the wrong action, set up control and prevention mechanisms based on faulty or incorrect data, then it doesn't really matter anything. So this is just some examples of a ECRA trial that we did last year here in Southeast Asia in relation to the UK Aid Fleming Fund ECRASIA project. So this is the data from 15 public health laboratories in this region testing into cocci. And as you can see, 2/3 of the labs have actually deviations above 5%, and some of them actually up to 30%, which is a bit scary when you think about that this is surveillance data, but it's also the labs that actually provide data for treatment. So if 3 out of 10 tests is incorrect, then there might be other solutions to this. So that was already thought into the surveillance regime more than 10 years ago. So I think it all started with the launch of the first Illumina benchtop instruments and the launch of the Global Microbial Identifier. So you might not have heard about the Global Microbial Identifier. That's actually a meeting going on right now in Vancouver in Canada. But it started with a meeting in 2011 in the EU Commission, where DTU, along with the US CDC and the US FDA, had a talk about how can we move a genomic-based surveillance forward. And here it was visioned that at that time, that in 5 to 10 years, we would actually have sequence instruments in most of clinical labs, and that the cost of a bacterial genome would be about €50 or $1, and that we would have the capacity to share data across borders in databases and being able to do jointly cluster analysis. That was kind of the vision back in 2011. So basically the concept of that was to move from single isolates with whole genome sequencing and then allow us with bioinformatic tools to identify species, resistant genes, virulence factors, and also cluster analysis. And that would enable us to detect emerging resistance, clones, in a faster pace than we do with phenotypic methodologies. So I was supposed to talk about new technologies, but I'm sad to say that the new technologies is already a decade old. So I think that nothing have really emerged since the benchtop whole genome sequence instrument. So I did a bit of timeline here where we can see that in 2010, there was the launch of the HiSeq, and more or less at the same time, there was also the launch of the MiSeq, which is a more medium level platform. And this was also at the same time when we had the GMI meeting in Brussels. And then, of course, there have been releases more or less every year, but again, it have not been major developments. So I just put in a few, so the NextSeq replacing HiSeq, and then NovoSeq, which is being used for really bulk analysis and more metagenomics. But we shouldn't forget also the ONT that there was a bit of a hype around in around 2010-11, and then finally it was marketed in 15. And that's opposed to Illumina, a long read technology. And that has actually some promising features that I will also touch upon. So what is also important when we talk about a genomic-based surveillance is, of course, cost. So it was envisioned that the cost would be like €50 already in 2011. So this is just an example of current cost. This is example from 19. And in one of the columns, we have Public Health England. And basically at that time, in 2019, the cost, including also labor, was about 125 euros. I think that was a bit conservative. I would think that we can actually generate a genome for less than 50 euro today, and with staff cost, it would probably be around 100. So we are not that way off with our vision back in 11. So what is new is not sequence platforms, but it's more applications and tools. So I did a few years ago, also in 2019, along with the US FDA, a bit of a review on what sequence tools or bioinformatic tools are available for predicting AMR. Since this is a meeting about AMR, I just list all of them. So I identified at that point 47 tools that can in silico predict resistance. So the question is how useful this is. And we really need to benchmark these tools, and not only now, but also in the future, because that's where the new technology lies. That is with new application, new tools. So we definitely need to ensure that with the launch of new tools, that these are proper benchmarked, because otherwise novel users would have no clue if where they can actually trust the outcome. Because what is difficult in a whole genome sequence-based surveillance is not to generate the genome, it's to actually analyze the data, because as also Dr. Shawar showed, that suddenly you have 1 million genes, and what does they mean? How do they confer resistance? And I also saw pictures or illustrations from Professor Park showing a number of genes, and basically a lot of people already have problems with determine what confer resistant to what. So it's not, I see it not every day, but weekly, that for instance, TEM1, people think that this is a resistant gene conferring resistant to third generations of response. No, it's just ampicillin. Then all the OXA genes, it's a mess. OXA10, they believe that that is a carbapenem resistance. No, it's also just plain penicillin. So again, whole genome sequence-based surveillance open up a completely novel area of understanding data. And on top of the resistant genes, we also have the mobile elements. So we talked about usage yesterday. And usage is definitely a driver, but there's also other drivers. There's sanitation, there's infrastructure, but there's also the contribution of transmission and mobile elements. So we shouldn't forget about mobile elements, because that is probably also an area that we need to pay attention to. So if you use a drug in the human sector, it can easily, even if it's not used in the animal sector, cause resistance in the primary production of animal and food. But anyway, all of these tools are useful. I have marked some in red that we have benchmarked, that we always tell people that they can trust. But some of these tools that you would find would either be commercially available in black boxes. They might also be open access. Some would take a lot of time to compute, others just a few minutes. Some would be lab-based, web-based, with open access. Others would be command line and pose a lot of complexities to the end users. But this is where we are. So where the problem also arises is still with the repositories. So when we generate data, what do we do with all this data? So we try to store them locally, but there's also a tendency that institutions, governments, regions do not really want to share these data, because suddenly they can be used for more than just the surveillance. So basically there's a tendency that we do not want to share data. I have listed here a number of surveillance programs that in one way or the other try to collect data and share it. But again, I still think that we are in its infancy. It's not really open access, even if those that develop these repository tell others that this is open access. No, it's not completely open access. It is with limited use. So I have experience with the ECDC platform EpiPulse, where you can see some data, but you cannot see all data. It's the same with Genome Tracker in the US. Again, some data is not shared. So I want to at least highlight a few examples here. So what I like is the FDA system with the Salmonella Resistome Tracker. I actually think that gives quite a nice feedback in terms of what data that are in the repository. It also allows for cluster analysis. Opposed to that, and I'm not saying that this is wrong, the other example is a European system where it's more a closed system where data are shared between organizations. So again, these systems all comes in different shapes with multiple purposes, etc. But it's not for all, and it's not completely transparent and open access, in my opinion. So as I indicated, that what I think is a nice system for now is what we have with the EpiPulse from ECDC. And again, it's not completely open access, but at least here, this is where we in Europe, where I have my best knowledge, is where we are able to share our Salmonella data and E. coli data and so forth, the genomes and the metadata. And there would be weekly alerts that be afraid of this emerging clone that have caused an outbreak of that, and so on. So at least we are going in the right directions. But again, I think this is an area that is also one of the limitations, along with a million other limitations to move this really forward. But at least, even if we have limitations and challenges, we should of course not be afraid of moving this forward. We have to ignore that there would be some intermediate difficulties, but if we don't push the boundaries, we would actually never end with the final result, which in my opinion should be a global whole genome sequence-based surveillance. I think that has so great potential that we need to overcome some of the barriers. So we have also tried with a UK Aid Fleming Fund program to set up surveillance in also Africa. So for those that do not know about the Fleming Fund, it's a grant of 235 million British pounds that it goes to low and middle income countries in Asia and in Africa. And it's managed by the UK government, and we have just seized the first phase from 19 to 21, and now moving into the second phase of the program, where there's allocated 210 million British pounds. But anyway, in this system, there are some regional grants. One of those are a sequence grant in Africa. So there's multiple players in Africa trying to set up surveillance. So this is just one of them. It has a funding envelope around 6 million British pounds. And the term of reference was to establish free whole genome sequence regional sites in East, West, and Southern Africa, and then within 3 years to sequence about 15,000 bacterial genome and do multiple training courses. And basically, we succeeded with doing that. We set up sequencing sites in South Africa, in Tanzania, Ghana, and expanding to Senegal, and but also pulling out from Nigeria, where we also had. So we ended up with 6 sites and are reducing now because of the funding envelope to only 3 sites. That was the term of reference. But we actually, in that period, succeeded to sequence around 20,000 bacterial genome and SARS-CoV-2. And we are also moving into setting up central sites, and I'm coming back to that. So basically, within these 3-4 years, we actually were able to establish some sort of surveillance in Africa. I know that this is not a full-blown surveillance system, but again, it's a proof of concept. It shows that it is actually doable to do this, to both collect the isolates, sequence it, and make sense of it and policy making. So what we want to do is take it to the next level, and that is to democratizing sequencing. So basically, when we have established a surveillance system with a referral system, we also want to see if what we collect centrally is also what we would see in frontline diagnostics. So again, the beauty of the long-read sequencing with MinION is actually that it's portable. So you can actually move it to the frontline clinics, you can move it into outbreak areas, etc. So that was what we also trained along with FAO recently in Ghana, where we hosted some training courses. And here, with the technology, we are able to multiplex up to 12 bacterial isolates on one flow cell and run it, and then being able to do the downstream analysis from a software that operates offline, meaning that you can actually take this laptop software, bring it to the field, do the analysis in the field, and then bring back your data, or when you have internet, to upload it and send it back to central analysis as well. So this is maybe where we see the future, and that is to do also a more centralized surveillance rather than have everything done in a reference center locally in each of the countries. So basically, what I think is novel in this area are the portable part of it with sequencing. So basically, this is just one example with the bento labs, that you can actually put a lab in your backpack, go to a site, and you do actually your DNA purification. You use your MinION, and then you actually sequence there. If you have internet access, you can also upload your data and do more in-depth analysis more locally. So that's one direction. The other one is that ONT have a tendency to produce gadgets, I will call it like that. So this is also just a bit of a disclaimer, but we discussed that a bit with Johns Hopkins University that have a bit of experience with these gadgets. So ONT have the idea of marketing in low batches new versions of MinIRON. And be cautious, they do not work as always. So it is maybe a bit of a fail and trial strategy. So again, now they try to market the Trax iron. I have heard, that's my personal opinion, that it doesn't really meet the benefits. But anyway, as I said, we have to move forward. So this is not a complaint, it's just a reality that there will be new platforms being marketed that might not work as intended, but anyway, we need to move forward all the time to make platforms better, faster, and more reliable, and at the same time try to build the needed infrastructure. So what is also nice with the MinIRON is, of course, also, as Dr. Chauvar talked about, that you can actually use it to close plasmids. So Chauvar talked about closing plasmids with PacBio, but actually you can also close plasmids, which is also where we're going in the future with surveillance. And that is actually to do hybrid assemblies with Illumina and MinIRON, and then actually being able to trace and investigate further the entire plasmids, as there have been several examples of in this conference as well. So moving a bit into another area of metagenomics. So I'll come back a bit to some of the examples of metagenomics, but before that, I just want to mention as well that when we talk about new technologies, we also need to mention the high-C sequencing. So again, this is a high-throughput genomic methodology that also embrace epigenomics. And basically, the idea of it is to build a more 3D genomic concept. And that, I think, has promising features. And that would probably close some of the limitations and gaps that we see when we do shotgun metagenomics in virus, where we can't tell really variants apart. They would be mixed up. And it would probably also close the gap that we have in AMR. So I have seen multiple examples of shotgun metagenomic studies. I've even been part of it myself. But there is a problem to link the AMR genes with the plasmid, with the bacteria. That is still one of the limitations. So even if we see very nice pictures and it gives some nice results, it needs still to be interpreted a bit with care. I think that high-C sequencing might actually close that gap in the future. I don't think it's just around the corner because everything is expensive, but this might be something that we should pay a bit more attention to. So there is a number of barriers, and I think that's really maybe why we haven't seen a lot of countries implementing whole genome sequence-based surveillance. Because as I said, we started 10 years ago, so why didn't it really happen? It didn't happen in Europe as well. So right now, we are modernizing with ECDC, the public health laboratories in EU with their diagnostics. So they are at the same level as in Africa, basically. And as also Ernesto talked about yesterday in the veterinary and food area, we were in the forefront maybe 5 years ago, but there was no funding in actually equipping all of the national reference laboratories for the food and the veterinary area, and because of that, we are falling back. And in our surveillance in Europe, it's only the specific surveillance of ESBL, AMC, and carbapemesis where there's allowed to use whole genome sequencing. The rest is still based on phenotypic testing. So we need to address these barriers. And I've just listed a few here that we definitely need the in-country buy-in. So governments need to acknowledge that this is a powerful tool and this is going to be the future. And we need the funding to make sure that we have established the IT infrastructure and the equipment in place. We need to ensure that there is a straightforward procurement procedures and there are reliable supply chains. I think Jeff can talk about all the problems about that when we try to procure ONT for Africa. There's a lack of isolates. So not all countries actually save isolates. They would collect them, and after testing, they would just discard them. So we need to save these, at least if we want to do single isolate sequencing. Then there's the trust and sharing of data, which is also one of the limitations of really moving forward. Staff retention. Especially in low and middle income countries, every time we educate and train a bioinformatician, the day after they have a job in the US or in Europe. Then there's the knowledge and training and education. Many countries do not have bioinformatic training or education. That is also needed. And then there's, of course, the IT infrastructure that we really need to invest in. So if we don't have this in place, we would never have a genomic surveillance-based genomic surveillance-based AMR surveillance. Anyway, I talked about metagenomics. So that is likely the next step, but personally I also think that it's a bit out in the future. I have seen multiple researchers talking about the global surveillance based on wastewater, on feces, field runoff. We even in DTU collected waste from airplanes in light of trying to do proof of concept of AMR surveillance. And I'm not saying that this is in vain. This is actually moving the technology forward, but we not just need to make sure that we interpret the data in care, and that maybe is not always the case. So I just have some examples here of a very fancy analysis I was part of myself, where we collected sewage from around the world. And again, we just need to make sure that when we set up something like that, that we think about what samples is it actually that we collect. So here we collected samples from wastewater treatment plants from industrial countries and compared with grab samples from developed countries, which would likely represent Mr. and Mrs. Smith and their dog, but it doesn't maybe represent the entire populations as the wastewater from industrial countries. At the same time, it's also a problem, as I indicated, to really piece everything together. But at least it gives indications, and I think that this is a great tool to be used as a screening process. So if you see signals in studies like that, you definitely need to act on it, and that might actually be to collect cultures and do more single isolate sequencing to follow up on that. So I'm not saying that this is not the future. I'm saying that this might lay a bit ahead of time, and it serve as a good way of picking up signals. So metagenomics is what we would call the new black. So it's really a hype. Everybody is talking about metagenomics. But we also need to bear in mind that when we talk about metagenomics, there's shotgun metagenomics and there's 16S metagenomics. And 16S metagenomics is a great tool to identify different species. So it has served a great purpose in COVID surveillance in wastewater, and it can also serve as a great purpose for identifying bacteria. But again, be careful when you want to piece again stuff together with bacteria, plasmids, AMR genes. It is more difficult than anticipated. You need a sufficient sequence depth and coverage. That might actually be covered by the high-C sequencing in the future. And then there's all around understanding the data. So this is not as trivial as it is with phenotypic data, where I think it's a bit more easy. Here we need to understand what it all means when we talk about mobile elements, resistant genes, and so on. And then when we look at these abundance profiles, we need to make sure that we also understand, so what do the cutoff means? So when do we see disease? When should we be cautious? and when is it just something that goes below the radar. So there needs to be much more research into this to set cutoffs so we know what actually we see. And then the problem is also cost. I think that metagenomic is about 2,000 US for one sample, so this is not cheap. So coming to an end, I think that in the last decade, we have seen a lot of whole genome sequencing moving into research and diagnostic. So again, new technologies, I think it's been there for quite some time, but at least we're moving into field and central site sequencing. I think it's a great opportunity and alternative to phenotypic suitability testing, as also Dr. Chawar talked about, that this has already been proven by research, and there's already enough studies showing the high concordance. There's a great added value in sequencing, so even if the cost is 100 euros or dollars, it would be less than if you do all the analysis with conventional microbiology. That would actually probably cost more with pulse field, plasmid screening, etc. And then we need to build the national capacities. It's been in academia for the last decade, but in national reference laboratory, it just started. And then we shouldn't be afraid of the risk of short-term challenges. We need to overcome them. We need to invest in that because that will be the future. And I'm certain that we need a bit more political investment and buy-in if we need to be prepared for the next pandemic, which would probably be with the use of whole genome sequencing, maybe with metagenomics. And I think I'll end here. So thank you again.
네, 감사드립니다. 그 질문하실.
So thank you very much. Do you have any questions? We will take some questions from the floor.
마이크 좀.
Thank you for your wonderful presentation. It was very impressive. It's not a question, it's a kind of comment. I don't know your background exactly, but I was very impressed. And international organizations like WHO or FAO should recognize what you have done and what you're looking for. In the future, how can we manage the MR all over the world correctly? So I think the international organization should collaborate and develop the kinds of future technique to disseminate all the member countries to develop and to find out how can we control AMR correctly. So thank you again, and in the future I hope that all the international organizations and each country may join your wonderful idea. Thank you.
Thank you, Professor Park. And I just want to acknowledge that both WHO, FAO, and WOHA are working along in these initiatives. So we are all well aware of it. I think that it probably need a bit more political buy-in from countries themselves, because again, I acknowledge that these health organizations do not have a ton of money to invest in this. So we all need to bring money to the table and the commitment, and then we might actually move somewhere. But it might be a good idea to bring it up in health assemblies. So I know WHO have their assembly, FAO have their, and again, as we had the resolution text, as you also quoted from 15 with Margaret Chan, we maybe need something around this as well to be prepared for the next pandemic using whole genome sequencing. So I think there is a need for the recognition of the power in the technology rather than the support because it's already there. But thank you, Professor Park, for the comment.
예, 혹시나 추가적인 질문.
Any other questions from the floor? If not, I have a question for you. So in our department of microbiology, microorganisms, well, when we isolate the pathogenic bacteria or germs, we analyze them and we do serotyping, and we analyze against antibiotic agents, and we are building up a database. Now, afterwards, in the future, the NGS technology will become more common, and we are building up a WGS-related DB as well. Now, at the government level, I don't know, we have some issues because we're working on 2 separate databases and we have limited budget. So ultimately, where should we focus more on? Which is the database that will end in the future? So we have DB on genotype and phenotypes, and we have research on the correlation between the two. And so in your opinion, where should we focus on? If maybe we can just choose one database and work on that, that might be better for us. So what do you think?
Yeah, very good question. So first of all, those that are moving into or replacing phenotypic surveillance and moving into genomics, there's probably a need, if they are accredited, to run the technology in parallel, and there's an ISO standard describing how to do that. So there needs to be an intermediate period of overlap to show that the concordance between it, unless that you trust the data that have already been shown, for instance, by the US FDA. In terms of databases, you're not the only one. I think that it's rare to hear some countries having only one database. So basically, even in Denmark, we have 3 institutions, one dealing with animals, the other one food, and then public health. They have separate databases, and that is because, again, the fear of sharing all data. So basically in Denmark, we have these 3 databases with joint interfaces, and that means that all of the 3 institutions can assess the genomes from all and do cluster analysis, and they would also be able to assess minimum data, metadata such as the species, the date, location, and source, and so on. But when it comes to privacy data, then it's the separate databases. So that's where they don't have access to each other. So I think it's good to have a joint database if you can convince all in Korea to have just one center, also to save money. I think that's recommendable. But again, it's more often to see that there are multiple databases in one country, despite of that.
네, 감사합니다. 예.
Yes, thank you very much.
좋은 발표를 해주신.
So once again, I would like to thank the professor for your great presentation, and we'll wrap up this session here.
네, 그럼
So this will conclude session 3. I would like to thank the moderator, Kim Soo-nam, director of the MFDS, and also the 3 speakers for their great presentations. Please give them a round of applause. Before having the next session, we are going to have a short break. We are 5 minutes ahead of the schedule, so we will have 20-minute break, and we will resume the session at 3:10.
안내 말씀드립니다. 곧 행사
Ladies and gentlemen, we will continue with today's program shortly. I would like to ask all participants to please come in and take your seats. Thank you.
your seat in the venue. Thank you. 안내 말씀드립니다. 다시 한번 안내 말씀드립니다.
Announcement. We are going to begin this session shortly. I would like to kindly ask you to take your seats.
지금부터
From now on, we are going to begin the final session of the third GCFA. Session 4 had 6 presentations. So they are going to talk about the ACT project of FAO. The conference is now live broadcast through the YouTube channel of MFDS, and presentation materials and also the video will be uploaded after the event. So for the session 4, we would like to invite the chair of the session 4, and I would like to hand over the microphone to the chair of the session. So Professor Park Yong-ho, and please welcome him with a big hand.
For your kind introduction. We are almost ready to wrap up through this last session of this conference. I'm very honored to be a chair of this last session, and then close, and then we can wrap up and make a final conclusion, and recommendations can be made through these sessions. Especially this last session organized by the FAO, future direction, and how can we solve the problem through the old efforts made from different international organizations. The first speaker of this session, Myung-Shin Choi, you already know much about her. She was with us at TFMR last 5 years. And then she is now the Food Standard Officer at FAO in Rome. Please join me. Welcome to Dr. Choi for her presentation the progress on the action to support the implementation of Codex AMR Act project.
Thank you, Dr. Park, for your kind introduction of me. Good afternoon, everybody. I'm Myung-Shin Choi. I'm the Food Standard Officer of the Codex Alimentarius Commission, and I'm coordinating ACT project. Before I begin, I would like to say my special thanks to the Korean government for hosting this important event and for your kind contribution to the ACT project and for giving us opportunity to share our experience of the ACT project. Session 4 is for the ACT project and is composed of 6 speakers. I'm the first one, and I will introduce the ACT project briefly, and then my precious colleagues will follow with more and in-depth explanation. I want to start with what the ACT project is important. ACT stands for Action to Support Implementation of Codex AMR Text Project. The full name is Implementation of Codex Standards to Support Containment and Reduction of Foodborne Antimicrobial Resistance. I would like to highlight 2 words, Codex and Foodborne AMR. It is the first project in Codex to support countries directly in implementing Codex text. And it is also the first project in FAO which is dedicated only to the foodborne AMR. You know who he is? He is Tom Hyland. He said this is the way it should be. I agree with him. This is brief introduction of the ACT project. It is a global project with 2 regions, 4 countries in Asia, Cambodia, Mongolia, Nepal, and Pakistan, and 2 countries in Latin America, Bolivia, and Colombia. We started this project in 2021 and will finish in 2026. It is a 5-year program. And the donor country is the Republic of Korea, the Ministry of Food and Drug Safety, with $10 million. The goal is to support implementation of Codex standards on AMR. Codex has developed relevant text to contain foodborne AMR. This is the heart of our work, compendium of the Codex text on AMR. It has 3 important texts: guidelines for risk analysis of foodborne AMR, and guidelines on integrated monitoring and surveillance of foodborne AMR, and code of practice to minimize and containing foodborne AMR. I believe this compendium will help countries in designing and running a successful strategies to contain foodborne AMR. This project has 3 objectives. Objective 1 is to raise awareness on foodborne AMR and Codex text on AMR. And objective 2 is to support integrated monitoring and surveillance systems of AMR and AMU in food production. And objective 3 is to strengthen the capacities to support good practice. This is our approach. We know that countries differ in their situations, such as socio-political context and level of implementation of Codex text. So, first step is to assess where they are, and then do the analysis, what their gaps are, and we plan the work plan. In this point, it's very important to prioritize the work because always we have a limitation of resources. And to the support capacity building, and at the later stage, we will assess the impact of the project. From now on, I will introduce the progress has been made so far. First, assessment has been made using FAO evaluation tools. The result, each country assessed that they were in the initial phase of AMR implementation. An analysis has been made and priority area has been found. We have found 4 priority areas. First, raise awareness of foodborne AMR to farmers, veterinarians, and government, and to improve collaborations between governments and the private sectors, to improve the national AMR surveillance systems through INFARM. My colleague Alejandro will explain it more later. Lastly, to strengthen the regulatory framework to stop using growth promoters. We also identified specific commodities of interest: poultry, cattle, pig, aquaculture, and others. Now we have a clear idea what we have to do. Our project is ongoing, but we have achieved something, so I want to highlight some achievement from now on. Objective 1 is related to raise awareness on foodborne AMR. Communication activities has been implemented to introduce the project and raise awareness of foodborne AMR and the codex text on AMR. Our project has been highlighted on national TV program, radio, and newspapers. And several dozen of news has been updated on different websites, such as Food Safety website, and Codex website, and FAO AMR website. And we also regularly update our news through newsletters. At the country level, they contribute various activities, especially during World Food Safety Day and World Antimicrobial Awareness Week in collaboration with the WHO and OA. I want to share a story from a small village, Hata Haital in Pakistan. Their main industry is dairy farming. The farmers followed household principles, and they used to believe antimicrobials are magic drugs that can cure every disease, but has not been the case always. She is Dr. Stamina. She's a veterinarian and has been practicing several years there and leading the campaign to raise awareness. And after farmers has changed their behavior, they are trying to follow good practice because they understand it is good for animal health, not compromising economic benefits. So this story will be posted on the web during the upcoming Antimicrobial Awareness Week. We have developed several materials. First, compendium. And we also have developed a brochure to explain what we are doing and what you have to do to contain foodborne AMR. Now it is available in English, Spanish, and Korean. I want to share a story from Colombia. Many agencies, including ACT Project, has been developing materials on AMR, but sometimes it is very difficult to find. So ACT Project in Colombia is developing an AMR platform in Spanish to collect information, to share and disseminate relevant information among ACT Project countries and beyond ACT Project countries. Objective 2 is related to support integrated monitoring and surveillance system of AMR and AMU in food production. I'm so proud of that. FAO has completed the development of in-farm IT platform, and Alejandro will explain it later. At the country level, they are currently in the process of establishing AMR monitoring and surveillance systems. Thanks to the ACT project, Mongolia is in the process of developing an AMR pilot surveillance system that has never done before. In Nepal, they are in the progress of developing an integrated surveillance system linking food and livestock production that has never done before. Objective 3 is to strengthen the capacities to support good practice. The ACT project has developed ACT 2 to understand the country's regulatory framework and level of implementation of codex text. My colleague Carmen will explain it more. And this tool has already triggered interest in other regions and will become available as global goods. Yesterday and today we have heard about growth promoters. Phasing out antimicrobial growth promotion in food is very challenging. The ACT project is developing resources to help countries to address the challenges of AGP removal. My colleague Jeff will explain it more later. This is a brief introduction of the ACT project. I want to conclude with this saying, no time to wait, act now to stop foodborne AMR. Thank you very much.
Thank you very much, Dr. Choi. It's a wonderful presentation. And then any question or comment? Floor is open. Okay. Please identify yourself and then ask a question, please.
전 식약처 이상욱입니다.
So I am Isanguk from MFDS. For developing countries, in order to reduce the AMR, I think there can be some things that can be done at FAO level, but in many areas, I believe that there should be some collaboration with the WHO and WOH. So for these collaborative works, how do you carry out them? How do you promote cooperation with other international organizations? So if you could give us that explanation, I will highly appreciate.
Questions. I briefly introduced the project activities, but at the country levels, they are always collaborating with the WHO and FAO. So maybe we cannot explain what they are doing in collaboration with the WHO and OA, but I believe they are doing, yeah. Especially for the raising awareness activities during World Food Safety Day and World Antimicrobial Awareness Week. Yeah, always they are collaborating each other, and they prepare the program together. And this is the way we should be. Yeah. Thank you.
Okay, Dr. Levana.
Thank you for a very nice presentation. I have a question. I think it's great to see all these examples of countries where nothing was happening in surveillance, bringing together data and starting to do an effort. I guess they do it also because they have help from the outside, and probably FAO and others are putting some funds on the table for them to start. Is there also an element of co-financing from these countries? And my worry is what happens once you go away from the country and leave them alone, do you think there will be sustainability of whatever they put in motion when they are left alone? Because I think that is a real pity if that is the case. So is there any medium-term or longer-term thinking about these activities? Thank you.
Thank you very much for your question. What we learned from this project is a 5-year project. We learned that 5 years is not enough for them to do the work by themselves. So first, I want to ask the Korean government for their continuous support until they can stand alone. And when we work, we try to give the ownership to the government, to the countries, especially government, because as you said, we live at some point. So every steps, we try to make them engaged on the work. Especially in Asia, there are some fundings. For example, Cambodia, they have flaming funds, MPTF, Multi-Donor Trust Fund on AMR, and ACT project as well. So in that case, we encourage them to plan together, not ACT project alone, because ACT project is based on what they have done, what FAO and WHO and WOA has done. So we want to complement, not fight each other. So when we work, we try to keep in mind to make them work together. It applies to our work as well. Thank you very much.
Thank you. It's kind of amazing situation, how the FAO Codex manages all different kinds of countries with a different level against MR. So I just wondering and then ask a question, how can you manage and then communicate with all different level of MR situation, all different countries and Southeast, the Mongolia, Pakistan, and Southern America? You have any special tools?
Thank you very much for your good questions. This is global project, so it is not easy to work directly with the countries. So our structure is like that. There is a headquarters and regional office and country offices. And we try to make a mechanism that enable us and enable them to work together. And first thing we tried to build was to make a stakeholder network at the country level and the regional level. So I'm so proud of that. 6 countries has developed strong stakeholder networks that enable them to work together. I'm so proud of that. Thank you very much.
Thank you. More questions? Okay. From OA.
Thank you very much. It's just a comment, actually. I just wanted to support what you have explained, everything, that we are collaborating on the ground with all the countries raised, and I can assure that that we are trying at the country level. We are together with the WHO country office and FAO country office, and OA doesn't have a country office, only a regional office. But so that's why I myself is actually the collaborator in the country level as well. So I can assure that we are trying our best as a tripartite and now quadripartite, that that the countries are involved in at the very early stage of this development of a project formulation, and we try our best to advocate for the domestic funding and sustainability. Of course, it's always one of the biggest challenge, I would say, but it's our mutual position that we promote the sustainability and country buy-ins. Thank you.
Thank you very much. Actually, not only the Republic of Korea, Japan is a good cooperator and assistant to develop this kind of action in reality. Thank you very much. Please join me to close the Dr. Choi's presentation. I'd like to move on to the next speaker, Andrés Garcia from FAO. He's a veterinary epidemiologist and now animal health officer. Now all yours.
Yeah, one second, because I made some changes. Yes. Thank you. I just have to put presentation mode, right? Okay, so thank you very much for the introduction. So I'm just going to show you some of the advancements we've made in the international FAO antimicrobial resistance monitoring IT platform and system. This is one of the key deliverables that are also under the ACT project. Is the mechanism we are also going to be using at the global level to support the implementation. I'm not doing this, but one second. Okay. To support the implementation of the Codex guidelines for integrated monitoring and surveillance of foodborne AMR. So hopefully we can bring more clarity on how we are going to articulate this collaboration and this mechanism for implementation at the national level. So just to start with a short introduction, something is wrong because I'm not touching anything and it's moving along. Okay. Just to start with introduction, so as you know, we are operating, all the FAO activities are operating under the FAO Action Plan on Antimicrobial Resistance. This action plan is covering the period 2021-2025, and this is the roadmap coordinating the efforts of the organization in the implementation of activities. So we have 5 objectives. Among these 5 objectives, one of them is strengthening surveillance and research. And I'm going to focus on this. For this, we have 4 main mechanisms to deliver on this objective. One is to deploy tools for capacity building on surveillance and laboratories in the field. We also do field interventions, collecting samples, doing antimicrobial susceptibility testing and pilot studies. We also provide access to resources and technical networks. And then one of the things that we are including in this new action plan for this, something is wrong. One of the things we are including in the action plan is strengthened support for data collection, management, analysis, visualization, and dissemination of the data. This is something new compared to the previous action plan, because now we are giving more emphasis to the use of the data produced from the laboratories. So we want countries to use this data for informing decision making. So of course we know integrated surveillance. We know integrated surveillance is recognized as one of the main mechanisms to support evidence in the One Health spectrum. So this is recognized, integrated surveillance in AMR, in AMU, but also integrated surveillance in humans, animals, and also the environment. So the quadripartite, we are working together in different initiatives on integrated surveillance. For example, just also to answer some of the questions our colleague was asking in the previous presentation. So we collaborate with quadripartite. We collaborate with different initiatives. For example, we have a technical advisory group on integrated surveillance that it was just established a few months ago, that they are starting to work on guidelines. But one of the initiatives that we are also coordinating with quadripartite is the Global Integrated System for Surveillance of AMR and AMU, which is called GISA. This is the mechanism that quadripartite agencies are going to be using to gather the information coming from the different systems and the different data that are collecting. So we have, for example, GLASS, the Global Antimicrobial Resistance Surveillance System, collecting information from AMR and AMU, that is going to be sharing this information with GISA in the future. We also have the Animuse platform. Our colleague Nahoko was explaining also on the Animuse platform, is collecting antimicrobial use in terrestrial and aquatic animals. And this information is also going to be gathered under this GISA platform. And under the responsibility of FAO, we have antimicrobial resistance in animals and food, and antimicrobial use in plant production and protection. And this is what we are going to be collecting through the International FAO Antimicrobial Resistance Monitoring Data Platform, the INFARM. So this is it was explained in the last year also, but I'm just going to briefly explain. This is the visual identity of INFARM. In the left, we have the data generation. So we have the bacilli and the cocci, the bacteria that are isolated. Yeah, something is not, yeah. In the left, we have the bacteria, that is the data generation part. In the center, we have the capacity building for transforming this data in information. So we want to transform data in information that is shared at different levels, and this is what we also want to represent in the right side. So INFARM is designed to support countries in collecting, analyzing, and using their data, but it's also designed to support countries that want also to share this information at the global level and want to share this information at the global level in the FAO platform, but also in the GISA platform. And this is what we will be supporting for countries. So when we talk about surveillance in food and agriculture, we always have different objectives. No? So we always talk about the public health objective. We also talk about the animal health objective. So these are the main objectives that we are doing that we have for antimicrobial resistance surveillance in food and agriculture. The one that we are going to be putting more attention and putting more focus initially, and the immediate priority, is to collect antimicrobial resistance data from foodborne bacterial pathogens and indicator bacteria. But the platform is designed to gather and to collect information from all different surveillance programs, also from the animal pathogens and from for animal health. So this is the main focus because, of course, this is also supporting the implementation of the Codex guidelines for integrated monitoring and surveillance of foodborne AMR. So understanding that countries have different levels of development of the, it moves alone, sorry for that, there must be a technical glitch, but it moves alone. So understanding that countries have different levels of development and implementation of their surveillance activities. There are countries that have national surveillance systems in place. There are countries that are just starting in some initiatives, like for example, Myo Singh was talking about Mongolia. They are doing this small pilot study. So understanding we have different levels, the platform is designed to collect information from both systematic monitoring and surveillance, but also from pilot monitoring activities. So the data is going to be tagged accordingly so that it's not mixed and analyzed together, but we are going to be collecting information also from these pilot surveillance activities. Then the platform is going to also be able to collect information on the context, information on reports, for example, the implementation of assessment reports, and also we have a questionnaire also to collect information on the different surveillance elements, different surveillance components in food and agriculture that is giving us an understanding of how this data was generated. Then in the center, we have the data collection, analysis, visualization, and interpretation. Countries are supported for these 4 functions, and countries can do this accessing to the private interface. There will be a private interface with restricted access in which countries can log in and look at their data, look at different interactive data visualizations, the dashboards that we are finalizing the development right now. And this is going to be migrated on an annual basis. There will be an annual publication cycle like there is in GLASS, like there is in other surveillance systems in AMR, that this data will be migrated from countries that allow to share this data at the public level, will be migrated to a global public interface. So just to give you some idea of the type of reports we are developing in the platform, we have reports that are produced automatically in 24 hours after the countries submit the antimicrobial susceptibility testing results from the lab in the INFARM AMR data files. We have reports that show the country profiles, so the volumes of data in the different bacterial species per specimen, also where the samples were recovered, also the type of surveillance program or objective and scales of monitoring. All the dashboards have in the right always different filtering options, of course, because the filtering options are essential to understand and to interpret the data. So this is another type of dashboard, which is showing also the volume of isolates, submitted in the system for different bacterial species according to the specimen, origin, etc. And this can also be filtered by year, scale of monitoring, interpretation criteria, etc., etc. Another example is the antimicrobial resistance prevalence, or the proportions of antimicrobial resistance to a specific given antibiotic panel. Here you can also filter by bacterial species, and you can also filter by interpretation criteria, etc., so that you cannot make misinterpretations. We also have analysis of trends. There will be a possibility to analyze the trends on resistance if countries want to monitor the progress in their interventions. If they are, for example, placing a ban on the use of a specific antibiotic, they can see if they is being efficient or not. And we also have multi-resistance proportions available in the platform that will be also having a longitudinal analysis per year. We are working now, finalizing the development. So as Miriam Singh was saying, we finalized the development of the beta version of the infant platform. And we are finalizing, with the feedback we receive from countries through the piloting phase, we are finalizing the final IT solution. In the final IT solution, we will include more dashboards. So far, we have 9 different types of dashboards with maps as well, maps with different color and different shades of color depending on the prevalence. We will also be adding a new dashboard on the distribution of minimal inhibitory concentrations, MICs, in tables, but also in graphs for countries that are using this methodology for antimicrobial susceptibility testing. So just some of the benefits for countries. So this is a safe place to store data with restricted access and possibility to share it at different levels of confidentiality. These are easy tools to analyze and interpret data. This is one of the main gaps we identified through many assessment missions we did in countries, is that countries produce data. Countries produce data even through pilot studies, but also through systematic surveillance, but they are not using this data. They might have like tons of Excel files and tons of data, but they are not using it for anything because they don't even, or maybe they don't have epidemiological capacities, or maybe they don't know how to do with this data, how to interpret. So we hope this new platform will also help countries to analyze, interpret, at least to produce some of the reports. Of course, this will never substitute a full epidemiological analysis of your information, but at least this is a first step for starting to use the data. Then it will also be a one-stop shop with repository of documents, information to contextualize the data, as I was saying before, and it will improve coordination in data collection, analysis, and harmonization in data generation. And I will explain a little bit more on that later, because there will be also a figure showing how the data is integrated across the food chain and coming also from different institutions. So this is hopefully going to be one of the solutions also for countries to share data between ministries, between different agencies. This might break some walls, I think. Then also will contribute, of course, to the global One Health AMR surveillance efforts. So just to show you the roadmap. So in 2022, we finalized the development of the beta version of the platform. Then in beginning of 2023, for 4 months this year, from January to April, we're having a pilot phase with countries that express their interest to participate. They were sending information on their surveillance systems, but also they were submitting antimicrobial resistance data to the platform. They provided extensive feedback in the use of the application. With this feedback, what we are doing now is to integrate it in a final IT solution. And so this is what we are doing now, expanding the functionalities of the platform. At the same time, we are developing guidance materials that will help countries to participate in the initiative. And in parallel, we are also building the interoperability with HUNET and other laboratory information management systems. This is very important because the management of the data, the cleaning, validation, management of the data is a step that we know is going to be a bottleneck for many countries. We want to facilitate the transmission of data from the laboratories to the platform directly. So, and this is going to be facilitated, like it's done for GLASS with HUNET, is going to be also facilitated for Infarm with this data management software. In 2024, beginning of 2024, we plan to launch first annual open call for data. We plan to start the global rollout of the initiative. Then, of course, there will be the publication of guidance materials. We will couple this with trainings in data management, trainings in HUNET, trainings in laboratory information management systems that are adapted to produce ready-to-send in-farm AMR data files. And then we will also couple this with additional IT resources. 2025, we expect this is the phase of expansion with the first global report published already, maybe in 2024, but might be in 2025 because things are sometimes slower. Then there will be a second annual open call for data on an annual basis, and we will start the cycle of annual data production, and then integration with other data platforms. This is also very important because as we are talking always about integrated surveillance, it will be important to start conversations with other agencies to see how we can do not just putting data in a global repository in GISA, but also how to start doing integrated analysis. So analysis like the one that was shown at the European level, for example, from Ernesto, from the GIACRA reports, the joint reports from the different agencies. 2026, phase of consolidation. We expect the annual open calls continue, but we also expect to expand to other programs of surveillance. We were discussing in the session before about whole genome sequencing, about next generation sequencing technologies. We understand this might be the future also for surveillance. We need to also start thinking on how to integrate this data also as part of the INFAM system. And of course, also antimicrobial use in crops. This is something that we are already starting collaborations, and we are starting also to expand knowledge on that, to understand what are the sources of information we can use to start producing some data on the volumes of antimicrobial use in plant production and protection. So as I said before, launching the open call for data, we need to produce guidance materials. When we talk about guidance materials, there are different levels of practical details we can give. So the codex text and the guidelines on integrated monitoring and surveillance are giving ingredients, but it's like the book of ingredients, but sometimes you don't know how to use these ingredients to make the recipe. So these are the texts, how to implement it practically. There are different guidelines, for example, in Asia Pacific, but in other regions, that are supporting the implementation of specific surveillance programs. Surveillance programs in healthy animals, in diseased animals. These guidelines are very useful for supporting the specific programs and are giving more details on, for example, how to calculate a sample size, what are the sites for collection of samples. They are giving more practical details. Then we also have protocols for implementation of integrated surveillance at a higher level of detail, like the tricycle protocol from WHO. And in the middle of this, at the level of detail that is between the ingredients, but also between the guidelines, we will have the manual for operationalization of INFARM and how it contributes to integrated surveillance. In this manual that is in preparation, we are including a framework for harmonization of the data collection, framework explaining the different surveillance programs in food and agriculture, giving some recommendations for which interpretation criteria to be used, which antibiotics to be tested for the different bacteria. We are also going to give some of the steps for participation in the INFARM initiative. So how to participate in INFARM, and just a very quick summary of some of the things we are including already in the manual. So to participate in INFARM, we have defined 3 simple but complicated steps. So it depends on how you look at that. So the first step is to identify the AMR surveillance components in agri-food systems. So we know that many countries are doing antimicrobial resistance surveillance activities, but they don't identify the different components across different ministries or across different institutions. So we want to set this as the first step for specific surveillance programs, but also for countries to start thinking about how to integrate these different surveillance programs. The step 2 is to identify the national INFAM focal points and start reporting. And countries report on 2 things. Countries report on the status of AMR surveillance in agri-food systems. So they have a questionnaire on the main components of surveillance systems, and then also reporting on antimicrobial susceptibility testing results from the laboratory, so AMR data. Then the step 3 is to commit to gradually establish the national AMR surveillance in agri-food systems that can gather and communicate on a regular basis information into the platform. And I'm going just to dive in some of these steps. So in the first step is to identify AMR surveillance components in agri-food systems. And when we talk about these different components, we have different surveillance sites. We have farms, we have slaughterhouses, we have places where the food is sold. So we have different, we also have diagnostic laboratories doing diagnostic of diseases in animals for animal health. So, and these might fall into different ministries: Ministry of Agriculture, Food Safety Authorities, Ministry of Health. So it's important that countries know what is going on in their country and what can also be designed in their country. So we have defined three main elements of let's say this is a national surveillance network, an ideal national surveillance network, but there might be different models. There might be different models of a surveillance network depending on the country. So the three elements of a surveillance network are the peripheral laboratories. So these are laboratories processing samples from the different surveillance sites, isolating the bacteria, and sometimes doing antimicrobial susceptibility testing, but not always. Sometimes these are the peripheral laboratories that are producing the volume of samples that are then sent to the reference laboratory. The reference laboratory is responsible in many cases for antimicrobial susceptibility testing, for confirmation of the results, validation and standardization of methods and results, and also the external quality assurance proficiency testing to ensure the quality of the antimicrobial susceptibility testing in the laboratories. Then we also have the National Coordination Center, responsible for coordination of surveillance activities in agri-food systems, the development of national surveillance programs, defining the objectives and designing epidemiological protocols, and also responsible for the dissemination of the information that can be externally through annual reports, but can also be internal through bulletins, or also sharing the results with the different stakeholders. So this would be an ideal network, so countries can identify which of the elements are in place. Sometimes countries have more elements than they think. When you go on missions, for example, for the assessment tool for laboratories and surveillance systems, we go on missions to countries, we realize that in the discussions with the different stakeholders that they have more things that they thought, or at least they have things in an embryonic stage. So they might have already some pilot activities or even research activities at the university. So this might be something that countries can start building on. Then the second step is to identify national infant focal points and start reporting. So the first thing is to identify the infant focal points. And an infant focal point are defined as in the slide. So these are nominated in coordination with the national bodies overseeing the development and implementation of AMR surveillance and monitoring in food and agriculture. And these are ideally experts with knowledge and experience in epidemiology and data management of antimicrobial susceptibility testing results. There can be one single infant focal point per country. This is a situation that we found in some countries where they have maybe just one surveillance program in place, but there can also be several infant focal points nominated for each of the specific surveillance programs. And when we talk about surveillance programs in food and agriculture, we divide it in 5. I'm going to explain later in more detail, but we have healthy terrestrial animals, diseased terrestrial animals, healthy aquatic animals, diseased aquatic animals, and food commodities. These are the 5 main programs that a country can define for surveillance. And in many cases, there might be several focal points responsible for reporting these different pieces of data on an annual basis. So the minimum requirement to participate is, of course, to enroll in the platform. The countries have an enrollment questionnaire. They send the enrollment questionnaire with the identification of the focal points. And once this is done, the first thing that they are asked is to complete a surveillance components and implementation questionnaire. This questionnaire is already integrated inside the platform. This is something that we received feedback that in the pilot phase. The pilot phase was a Word document, but now it's already inside the platform. Countries can complete this questionnaire. We can also get all the answers in a database for a further analysis. And this surveillance components and implementation questionnaire is going to identify the main different elements of the surveillance system. It's also going to identify if they are doing a quality assurance of the samples. It's going to bring some context. It's not like a full Atlas mission, a full antimicrobial, so the tool we are using for antimicrobial resistance surveillance capacity building is not like a full Atlas mission, but it's something like a small version of the Atlas tool that can serve to identify and to contextualize the AMR data. then countries can, the second thing countries can do, so the submission of the surveillance core component questionnaire is something mandatory for participation. And this is something, of course, that is desirable, that countries also submit antimicrobial resistance data. So this is is not a requirement for participation in the initiative, but countries, of course, are going to be invited to submit antimicrobial resistance data into the platform and to submit the antimicrobial susceptibility testing results. For this, there is a data model already specified. The database is already built upon this data model that is harmonizing the data collection, the different attributes, the different codes that are recognized by the platform. And this is in Excel, but it can also be submitted in comma-separated values, different formats. And with the collaboration we are establishing now with HUNET, it can also be produced automatically for sending it to the platform. And then this is something that will help also in this process. So the process of identification of infant focal points, but also the process of reporting, the process of collating the information on an annual basis and putting it together is going to be done according to this framework. So we have developed this framework, which is relatively simple, but not always understood by all countries, in which we have different steps in the food chain, so primary production, processing, and consumption. Then we have different surveillance sites associated to these different surveillance programs. As I said, we have 5 surveillance programs, and then in these 5 surveillance programs, for example, if we focus on the left side, this is for the purpose of animal health. We have the diseased terrestrial animals and the diseased aquatic animals. This is basically coming from veterinary diagnostic laboratories that collect samples from diseased animals, from bacterial infections in animals, different specimens, and different animal pathogens in these animals for this specific program, the specific program of diseased animals. Then for the healthy terrestrial animals, and for the healthy aquatic animals and the environment, and for food, we have common indicators that are defined universally, the common indicators, commensal indicators, Escherichia coli, Enterococcus, but then also the foodborne pathogens in terrestrial and in aquatic animals. And these are common for all the, for the public health objective, of course, and for these different surveillance programs that I was talking before, so for the food commodities, but also for healthy terrestrial and aquatic animals. As you can see, this is covering the whole food chain, and is covering the different steps in the food chain, also the post-mortem stages. And this is important so that countries understand what is the type of data, the type of information that is going to be collected through the platform. And of course, this has an impact also in the nomination of infant focal points. And I'm just going to show you 3 different scenarios that we identified through the pilot phase. Through the pilot phase, we identified that sometimes we have one focal point covering all the antemortem phase, so covering all the live animals programs coming from the Ministry of Agriculture. This focal point is responsible for submitting information from diseased animals, but also from healthy animals. And then one coming from the food side, coming from Ministry of Health, in many cases, or food safety authorities. This is one scenario, but we also have other scenarios in which Ministry of Agriculture is responsible for everything that happens until consumption. Then consumption is just under Ministry of Health in some countries, and they can report data, for example, passive surveillance in foodborne outbreaks. This might be something that they can also report on the platform in Salmonella and Campylobacter, for example. Other situation is a complete separation in the side of veterinary diagnostic laboratories in the diseased animals. So we have many diagnostic laboratories that are responsible or separated from the healthy animals, from the healthy aquatic animals and the active surveillance programs. So basically all the diseased aquatic and terrestrial animals, this is mostly done through passive surveillance in clinical samples. And on all the right side is mostly done through active surveillance, except the programs for foodborne outbreaks in consumption. So to report the data, to report the AMR data, understanding this framework for surveillance, countries at the infant focal points are responsible to put all the data together in in-farm AMR data files. And what is an AMR data file? What defines this entity. This entity is defined by these three attributes. So it's an INFARM data file is defined by the combination of these three attributes, so year of sample collection, level of representativeness of the data, and the surveillance program that we were talking about before. So, for example, one INFARM data file would be from year 2022, from a pilot surveillance activity that is representative only at the local level, so it's not having a representative sampling scheme, and from healthy terrestrial animals for public health purpose. This could be one single INFARM data file. The countries are responsible for putting these different data files, and then they can decide on two things. They can decide to report through different model options, option A and option B. Option B is reporting antimicrobial susceptibility testing results at the isolate level, disaggregated, and option B is reporting it aggregated, like they do also for glass right now. Then countries can decide on the level of confidentiality of this information gathered from this specific file. And there are three levels of confidentiality. I'm going to explain them now. So the first level is to keep the information private, only for the country. So countries are going to share the data into the private interface. They will access the interactive data visualizations just for their own. And then there are two public levels that countries can decide to share this information. The public level 2 is aggregated, so countries agree on sharing this information in the private interface. But on an annual basis, we will migrate all this information to the public interface, but without showing the country identity. So we are going to make the country identity blind so that we can also show the information. This is something like WOHA is doing now at the regional level with the Animuse platform. And then the public level 3, which is the most desirable one, the one that is used in many countries, but also in the European Union. This is the one that they are using also in glass, is use this level of confidentiality. So countries share the data into the private interface. They get access to their own data visualizations. But then on an annual basis, we will migrate all this data to a public interface, also showing the country identity. And of course, this is the one that we have established this hybrid modality of levels of confidentiality, also with the hope that countries gradually move towards level 3. And yeah, this is something that we will need to work over the years on how to do that. Then something that is important also in this step 2, when we need to report data, as I was saying before, is one of the bottlenecks is to manage and clean and validate this data. And we have now just signed an LOA, a letter of agreement with HUNET developers, and they are going to work on the interoperability of INFARM according to our data model, and also they are going to develop their tutorials and their trainings for exporting data to the INFARM platform. So this is very important because it will be the mechanism that many countries will be using for managing the data to submit to the platform without doing all this manipulation of data in Excel files. So hopefully this can solve some of these bottlenecks. Then we go to step 3. So we identified the surveillance components in step 1. We then identified the focal points. We started submitting information on surveillance, but also antimicrobial resistance data. And now the step 3 is to commit to gradually establish this AMR surveillance in agri-food systems. And for this, FAO has the assessment tool for laboratories and antimicrobial resistance surveillance systems. The FAO Atlas is quite a mature tool already that has been deployed in more than 55 countries and 200 laboratories. We have community of assessors in Asia, Africa, and Latin America, and it's a tool that is helping countries to prioritize actions to move to progressively improve their capacities for laboratories, but also for the surveillance systems. So the tool is giving a stage from 1 to 5 to the capacity of the laboratories to do antimicrobial susceptibility testing and bacterial isolation, but it's also giving a stage between 1 and 5 for the whole surveillance network. So according to this, the next step, and this is what we are initiating now with our information technology division, is to digitalize Atlas. Atlas has been a tool that has been always Excel-based. Excel tools are very difficult to use, are also very difficult to analyze. Many countries and many stakeholders are always asking, where is the Atlas tool? Is it published? Of course, we can publish an Excel file, but in the end, the use is not going to be the best if we just publish a very, very long questionnaire in Excel. So what we want to do is to digitalize and to provide access through a digital solution that will create automatic visualizations, but at the same time, will also give us the opportunity to have all this data in a database that we can analyze and that we can also produce information. Of course, Atlas is a quality check for data in the end. So countries receiving stage number 3 in their data generation network, but also in the surveillance network, we can rely on this information. We can say this information is more reliable than a country at level 1. And of course, this is also very relevant for the platform because it will be the quality check for the data we receive into the system. And just to close, so FAO is contributing to surveillance and monitoring in all these different steps. So we are providing support to countries in the overarching design, so countries to design their national surveillance programs, but also we are supporting countries in the data generation and management of the data now with this HUNET letter of agreement just signed. We are also supporting countries now with the Inform Data Platform for the analysis. And in the left side, we have all the production of data. But the most important thing is to transform this data into information. Transform in information, in findings, evidence that is supporting policy recommendations to support all the different interventions that we can do in the fight against AMR. And with this, I would like to close the presentation. Thank you very much.
Thank you, Alejandro. Actually, we are quite behind of the schedule. However, I'd like to invite any comment and question. Okay.
Thank you very much, Alejandro, for an excellent presentation. You have all my admiration. I cannot even imagine how difficult it must be to bring all this together, knowing how difficult it is only in Europe. Maybe one comment there. You showed this letter of, or this memorandum of understanding with HUNET, but perhaps one of the obstacles that you will have is those countries that are already collecting data and reporting data to other databases. So have you thought not only about this particular link, but how to provide help to those that need to convert their data models to feed your system? Because if a country needs to report to 3 or 4 different places, I think it will be quite a challenging thing. That's the first comment. And then a question. Have you done any kind of pre-starting questionnaire about these confidentiality levels and how many countries are already prepared from the beginning to go to level 3? Thank you.
Thank you, Ernesto. Very good questions, because on the first one, on how to support countries that already have national surveillance systems in place, high volumes of data, like the European Union, like the US, like Canada, like many other countries that have public data in different repositories. This is something that we need to think further. We think that HUNET is going to solve part of the problem because many countries are already using HUNET. But I think in the specific case of the European Union, we might need to go through memorandum of understanding with these members, specifically, because I understand now EFSA is collecting information from individual countries, and then you share this information to the European Union as an agency. But of course, we can go country by country, but it would be more efficient just to do it through EFSA, for example. So countries have an agreement that if everything they share with EFSA will be collated together in EFSA and will be shared commonly into the platform. So this is something that we could discuss, and this is something that would be super interesting, because I think the European Union would be could be also a beneficiary, because in the end, all countries are going to be able to see the different prevalence levels and to make comparisons, not for the bad purpose, but for the good one, for the purpose of advancing together. With other countries, we know that the adaptation of the data models is not that difficult. For example, Canada, and I can say this publicly because Canada was one of the countries that was also has public data. All the data is public in the CPARS program. They were also sharing the data in the platform, and it just requires, of course, someone, an epidemiologist, doing the transferability of the different codes and the different attributes. Of course, it takes some work, but once the code and the syntax is written, this is something that just needs to be run on an annual basis. Of course, yeah, it's a barrier. Yes.
Thank you.
Yeah.
Okay,
And well, maybe for the second one, the second one was on if we have an indication of which countries are going to be sharing at which level. Right now, we don't have this indication. We have the indication that countries that already have public reports will be happy to be at level 3, of course. Yeah, but in low middle income countries that are just producing initial data, we don't have an indication. And I think this will come in the global rollout. Yeah.
Okay, I want to invite Nako. In your OI experience, do you have any recommendation, suggestion to establish in-farm, that program? The microphone.
No, I actually had a very specific and practical question. How are you going to manage the access to this platform by the countries? Because especially when you expect that multiple people would access from one country, and when these people are not affiliated to the ministry directly, so that practically speaking, for example, because I know the faces of those who may access to this in this region, right? So, for example, some laboratories experts not affiliated directly, and when they are handing over their roles, responsibility to their successors, etc., we can imagine all this would happen soon after you roll out the database. And I wonder, or probably because we are in a very similar situation as I just said. So maybe we can exchange those practical, experiences and questions together to really run the database effectively together.
Yeah. No, that's a very good point, and thank you for raising this question because it's one of the key points. We have regular conversations with WOHA, so we've been discussing many times also with WOHA headquarters colleagues on their experiences. And in the end, the main solution here is to nominate official focal points. So that's why the nomination and the access we provide to the platform is going through ministries. So when we invite countries to participate and we will launch the global rollout, we will invite ministries of agriculture, ministries of health, and food safety authorities. So we will need to reach all these channels. And it will be the ministries that need to nominate the focal point. Of course, they should not nominate 20 focal points from the laboratories. In the end, the idea is that the platform is for national and government use in the end. So it's not for laboratory personnel on a specific surveillance program, because what we want is to collate this information from the different laboratories in one single place.
Yeah. Okay, one more question. Then we'll close to next presentation.
Thank you. It's a comment and also to share the experience. I think that we need to talk in the quadripartite, right? Because we have the same experience with glass. And exactly for the European region, we have an agreement with the CDC because they are also publishing that. So we get the information that is published in glass through ECDC. So from the European regional office, we get the information. This is one. The second one, this is question, it's important to also think about the quality of the data that you are going to receive, because the countries can be interested and they are collecting data, but we talk about the quality of the laboratory susceptibility testing, right? You can receive many salmonellas with high resistance of carbapenems, and basically it's because the antimicrobial disk is not working. So something like that, I think that you need to consider, because it's a lot of information that you are going to receive. And the last one, we were discussing the last point. Okay, so we will talk later. But Congratulations, because I think that is really good initiative, and it's the one that we need to finally have information from the different sectors on
Yeah.
AMR.
Yeah.
Yeah.
Okay.
Yeah, it's about the focal points. The
Okay.
focal points also, we have an agreement with the countries, and they designate in the case of glass, from the Ministry of Health. But
Exactly.
I understand that. But it's also important, I mentioned in my presentation, the AMR committees, the national AMR committees, because it's where all the different ministries are working together on AMR. So I think that is the place to decide who is going to deal with the data from each sector or one focal point for all the data.
Yeah, yeah.
Okay, he mentioned about that the National Coordination Center is important to validate all the data, to transfer that different countries to make a national action plan, followed by regional, and then finally global action plan.
Yeah.
Thank you very much.
Thank you.
Okay, let me move to the legal point. Next presentation, the speaker is Carmen Blon, the legal officer of the FAO. Oh, thank you. Thank you very much. Carmen Blon, Caro is attorney and law major. She is going to talk by Zoom connection, I believe. So please join me. Welcome to Carmen Blon for her presentation. Okay. Carmen Blon, all yours.
Thank you.
Can you hear me?
Thank you very much. Yes, I
Okay,
can hear you.
go
Good
ahead.
morning. It's really a pleasure to be here with you today. Let me just check if you can see my presentation. Please let me know when that's the case and if it is in presentation mode. You cannot see it? Okay, I'll try to share it again.
Okay, just a minute. Okay.
Can you see it now?
Okay.
Okay, perfect, perfect. I hope when I will move from one slide to the other doesn't take that much time. But anyway, thank you so much. My colleagues have greatly introduced the purpose of the project, of the ACT project, and the different work streams. So what I'm going to present you today is what we are doing in the legal component of the project, which is one of the components that's focusing on supporting countries, strengthening the regulatory frameworks to better support the implementation of Codex standards on AMR. So first of all, yeah, I'm sorry it takes a moment to change
슬라이드
slides.
공유가 안 되는 것 같아. Okay.
Here
Okay.
it is.
Okay.
Yeah. So first of all, how can national legislation, regional legislation support Codex standards implementation? I mean, it's very important to understand that legislation is a useful tool for implementing AMR related objectives because it can serve to introduce concrete obligations and responsibilities for governments, for business, and for individuals. But legislation is also important to set up institutions and to crystallize integrated governance and surveillance mechanisms. So for these institutions or for these mechanisms to become compulsory so that the different actors are subject to concrete obligations, it's important that there is a legal framework that gives basis to these mechanisms. It can also serve to regulate the sale and the access to antimicrobials. For only legislation can, for instance, prohibit the over-the-counter sale of antimicrobials. It serves to authorize, restrict, or prohibit the use of selected substances, and also to regulate obligations related to data and information sharing and to data confidentiality, and to introduce requirements for the production and disposal of antimicrobials and antimicrobial waste. So as you see, legislation is a very important step for countries to incorporate the recommendations in Codex standards into their national systems, into the national frameworks, and to make them not only compulsory, but also sustainable in time. And in this understanding, the project includes a legal component which basically focuses on two different activities. The first activity is the development of a global tool to monitor implementation of AMR relevant Codex standards into regional and national legislation. This tool is not only a legal tool, I'll talk about it a bit more later. It's a comprehensive tool that monitors the implementation of Codex standards. And then we have a second activity where selected countries' regulatory frameworks are analyzed, assessed, and strengthened in line with the Codex Code of Practice. So by supporting the assessment of the national legislation, by using the tool, countries also get recommendations to revise and strengthen their national legislation. And to do all this, we are working with different international consultants, including two legal consultants and a veterinary consultant who is an expert on participatory processes and co-creation processes, plus with national legal consultants in the different countries of the project. So let me now tell you a little bit about the tool. The ACT tool is an assessment tool. It's based on the Codex AMR Code of Practice, and also the guidelines for surveillance. And basically what we have done is to translate Codex recommendations into questions, doing an exercise of prioritization to make it useful and more focused, and trying to identify which of the recommendations in the Codex standards would require either legislation or policy, or national policy, to be properly implemented. So for each of these recommendations that would require legal underpinning, or that would require some national action, there is a question. The 80% of these questions focus on the legislation, with a 20% focusing on surveillance and other technical aspects. And the application of this tool, the tool has been applied, has been deployed already in the 6 countries of the project. And for that, we engaged with a national lawyer, a national legal expert, who helps us collecting the legislation and liaising with the national authorities. And as you can understand, and AMR being a multifaceted topic, which falls within the remit of different ministries and authorities, this has been a complicated exercise. So these consultants have been not only revising the national legislation, collecting and analyzing the national legislation against the tool, but also meeting all these different ministries, first in bilateral meetings, and then in plenary, all of them together, so that they can all listen to each other and identify where are the gaps and where are the issues that could deserve, or that could be opportunities for regulatory reform. And the outcome of these exercises is a short report with a snapshot of the national legislation, the identification of the key gaps, and recommendations for legal reform. Just to give you some examples of our preliminary findings. So for instance, in Latin America, in Colombia, we found that there's a legal issue with the sharing of data across different ministries that's hindering the possibility for these ministries to set up an integrated surveillance mechanism together. So because in the legislation there is no specific requirement for these ministries to share the data, they find they are not entitled to do so. And this is really creating complications at the time to setting up an integrated surveillance mechanism. In Bolivia, one example is that there is no legislation, there's no provision that introduce any sort of restriction for the use of antimicrobials as growth promoters. So this means that in the absence of legislation on this, farmers basically can do whatever they consider appropriate, and it's perfectly fine. Without legislation, there is no mechanism for the government to restrict the use of these substances. In Pakistan, we found a similar situation where there is no specific restriction or prohibition. There is no regulation on the use of antimicrobials as growth promoters. In Nepal, for instance, under the standard for poultry feed, the addition of feed supplements and feed additives in poultry feed is permitted, but the use of antibiotics is strictly prohibited. And we identified a problem with the implementation of the legislation. Also, another example, the Livestock Quality Management Procedure of 2016 restricts the recommendation of antibiotics for use in animal feed, but it allows for the import of specific oxidiostats. And so there are some countries where there is some legal basis, written in the law, but where this is simply not implemented. This is, for instance, the example in Cambodia that you see the diagram. The diagram is divided. I'm not sure you will be able to read the small letter in the circle, but basically these are the different topics that are addressed in the tool. Where the green correspond to those topics where most of the recommendations are incorporated into national legislation, and the reddish oranges are those topics that where there is less incorporation into national legislation. So, for instance, here in Cambodia, we have identified a gap in relation to prescription requirements. There is no provision requiring that only authorized professionals, veterinarians, can prescribe antibiotics and other antimicrobials. And there are no provisions that require veterinary medicinal products and other antimicrobials to be administered by a veterinarian or under the supervision of an authorized professional. So we identified that there is an opportunity to strengthen the legislation and introduce some changes. In Mongolia, you see the situation is a bit different, where and with no provision requiring that only authorized professionals can prescribe antimicrobials of certain types, but also the part on prescription and sale is pretty red, and no provisions regulating the use of antimicrobials for growth promotion. So we also see a potential opportunity in relation to the prudent use. And so this is the work that we have been doing so far. The reports are now being finalized, and they will be shared with the national authorities, trying to identify with them which are their priorities for legal reform. But we have started already thinking of what could be done next, bearing in mind that our tool is divided in the different chapters of the Code of Practice and the guidelines for integrated surveillance that you can see there in your left hand side. And we have identified that some of these topics, particularly everything that deals with institutional coordination, integrated surveillance, but also the access to antimicrobials and the prudent use of antimicrobials, we have identified that in these topics, countries do have significant gaps. I mean, it's not that there are no gaps or opportunities in the other topics, but these are topics where it's the different activities of the project providing good opportunities to move forward with legal reform processes. So what we intend to do next is to discuss with countries which are the policy priorities where they would like to be focusing on to try to trigger these processes that would lead to strengthening the national legislation. At the same time, we realized that enacting legislation is not enough for the legislation to be implemented. And that by, sorry, you can't see that slide. Can you see it now? Yeah. Anyway, you can see the different topics that we're addressing in the tool and how they correspond to some of the focus areas that are being presented to countries. So as I've said, I mean, the development or the revision of legislation is not enough to drive change. So what we are also working on is on methodologies to promote participatory approaches where farmers and different stakeholders are engaged into the processes of legal reform. And we have started doing this as a pilot through what we call a co-creation process. So basically a co-creation process is slightly different from a traditional participation process in the sense that it doesn't focus on all the different stages of participation. It focuses more specifically in the participation stages that would engage farmers directly in the identification of potential regulatory strategies to develop and to implement the legislation. So very basically, and in a manner that's understandable for everyone, what we do is to try to promote a dialogue between the farmers, the private sector, and the public sector, the regulators. This dialogue can take place for regulatory issues, for legislation that's applied at the federal level or the central level, but it can also take place for issues, for instance, hygiene improvement or biosecurity in the farm to reduce the need of antimicrobials that are taking place at the local level, at the decentralized level. So by creating these opportunities for dialogue between the private sector and the public sector, we work with countries in tailoring their national legislation to the actual capacities of the country to implement that legislation. What we try to achieve with these processes is to avoid enacting legislation that looks very good in paper, reflects very clearly what's written in the international standard, but it's very far from what can be implemented in the field. But also, so apart from giving us the possibility and the opportunity to developing tailor-made legislation that can be implemented, it also helps us building consensus in the society around which can be the best regulatory solutions to implement the legislation. And these are some examples of a recent pilot of a co-creation process in Cambodia, where we got very powerful feedback from farmers and different private actors on how useful it was for them to start understanding a bit better the reason why some of the restrictions have been incorporated and how to address them and how to find alternatives to the use of antimicrobials. So this is all from my side. It's really a very fast snapshot. I'm available to respond to any query you may have or to give you more information if you are interested.
Thank you, Carmen. Yes, the legislation is quite crucial and important to implementation of code of practice and the guideline of integrated AMR surveillance monitoring program. So is one quick question, then we can move to the next presentation. Naoko? Okay, you are the one.
Thank you so much, Carmen. It was a great presentation. I really, really appreciated the component that you presented regarding the participation of the community, the stakeholders at the ground level. because it's really, as you pointed out rightly, it's really crucial that not only the legislation is done, but it's enforced and implemented by those people on the ground. So I think it's great that you included that part in your tool. Then my question is, so after this review is complete, under this ACT project, do you have any activity to support the updating or maybe additions of the current legislation? If yes, that's great. If not, I just started to think, because you know this very well, Professor Park also knows very well, but just for the others, we have, WAA has a so-called PVS pathway, Performance of Veterinary Service pathway, and under that, we have a specific targeted support also. So that also, one of the package includes the legislation support specifically. So if this, once the reviews are done and if the countries want request us, I'm sure there will be a great collaboration, opportunity for the collaboration. It's great for the countries to really take up the result of this review, legal review, and put it into the actual legislation, update of the legislation. So I would love to continue to discuss about this opportunity. Thank you.
Thank you. It's not a question, it's a kind of a comment. I believe the OHA can hire you with a better position in the future for your utilization. Okay, thank you very much, Carmen. Okay, now we move to the next speaker. I'd like to introduce Jeffrey Lejeune. It's my great colleague. He's a food safety officer. Everybody know, I believe. He got his PhD at the veterinary medicine in Washington State University in Pullman, Washington. Because he is the same alumni, I got my PhD at Washington State University. So I'm very glad to introduce you as the next speaker about the practical approaches. Thank you, and then all yours.
Thank you very much, Dr. Park, for that introduction. Thank you for inviting me, and thank you all for sticking it out and hanging out here. I recognize we're on a little bit of a time crunch, so I'll try to keep my comments brief, but make sure I explain what I came for. Brief, I'll give you a little bit of historical background, kind of a justification for what we're looking at here. So it was in 1928 that Fleming discovered penicillin. And it was quite still a few years later after that before it became widespread use, okay? The first use was actually not in a child, but in a woman to treat a septicemia, I believe, some 14 years after that. If you go further, 1945, it was 17 years after his discovery that Fleming received the Nobel Prize for that work. And during his Nobel Prize acceptance, he made this comment, okay? He said it's not difficult to make microbes resistant to penicillin in the lab if you expose them to conditions that are not sufficient to kill them. So this is 78 years ago. This was a long time ago. This was even before Dr. Park was born, that this information was made available, okay? So we knew something was coming, right? That number there was 1942. They're treating people with penicillin to make them better, to cure illnesses and disease. 1946, just like 4 years after, they discovered that if you fed these drugs to animals, they got bigger, okay? So it became 40s and 50s, became a boom for providing subconscious lethal concentrations of antimicrobials to livestock to enhance growth, okay? 1951, so 42 and 50 was the years that they started using. 51, they start reporting resistant organisms, isolated from turkeys, from chickens, from pigs, okay, 72 years ago. So we know this, any antimicrobial use drives antimicrobial resistance, okay? Jump forward 54 years ago, 1969. Now, this is almost as old as I am, but not quite. The UK had a report that came out, the SWAN report. It was kind of a seminal report in antimicrobial resistance. And they came to two conclusions. One, well, they came to many conclusions, but I draw two conclusions here to point out to them. One was, look, feeding antimicrobials to animals, okay, results in selection and can pose a hazard to human health. Okay, 54 years ago. The other thing they say, antibiotics that are going to be used should have little impact. So you should assess what the size of the impact of the antimicrobial use is to determine whether they should be available for human use or for animal use or not. Okay? 54 years ago. We continue to feed subtherapeutic use doses of antimicrobials to animals. And finally, 2021, you've heard this before, you've seen this, right? A hallmark modern conclusion. Two conclusions here are principles. One is just the definition of growth promotion, which from this Codex document, very similar to what we see in WOA's documentation. FAO has a definition as well. It's quite similar. And it might even be in the MIA. I'm not sure as a definition. Two principles. Use of medically important antimicrobials is not a prudent practice, okay? And that should not be done. Does this look at all familiar? Kind of looks like these two conclusions they drew 54 years ago. I'm not trying to knock the Codex process. I'm glad we got it updated. But this is not new. I mean, Rene said he was going to not tell us anything new from 10 years ago. I'm not telling you anything new from half a century ago. All right? Antimicrobial use. So we need to do something about it. So if we've known this for so long, oh, here we go. Why are we still doing it? And you've seen this slide before too, okay? How much of antimicrobial is used in different countries, reported to the WOA through their self-reporting date, and how many of those countries has it legally versus a lack of legal use, okay? Now, Nahoko, can you cover your ears for a second? No, this is not any slight to WOA. They collect the data that they get, okay? From this slide, I would probably believe this one right here, this one bar, okay? The countries that use antimicrobials that either do or do not have legislation, okay? Over here, sure, you have legislation. I've been in many countries that have legislation, and they're pumping the antimicrobials into the feed to make the animals grow. So we don't really know. We're hoping this trend is changing, and we're hoping they're reporting fairly, and that's what we want. But we got to be, the issue is it's still happening, okay? And so this is what we need to address. Why are they still doing it? Well, maybe before we get there, what is the consequences? Now, we heard the other day, someone talked about the host specificity of bacteria, but people get sick from food contaminated with antimicrobial resistant organisms. That comes from animals. And this is part from a paper. Alejandro was a co-author on this paper. Oops, I didn't mean to do that. So here they were tracking a specific extended beta-lactamase resistant clones of a bacteria and kind of attribute them to source attribution based on their on PCR sequences and whatnot. Sorry about that. What I just want to point out here, so we have a lot coming from the community, so person-to-person transmission is important. But if you look down here, seafood, bovine meat, turkey meat, cattle, pork, some people eat horses, chickens, chicken meat. So we do know that there is this transmission. there have been outbreaks of antimicrobial resistant bacteria caused by food. If we look here, another a graphic from that same picture, here we have kind of the prevalence of these ESBLs in these different populations, which is variable, but was key. If you look here, this is like the percentage of the population that is exposed to a specific source, okay? And if I'm getting this wrong, Alejandro will clear it up afterwards. The idea is that raw vegetables, a lot of the population is exposed to raw vegetables. And raw vegetables are fertilized with raw manure a lot. Seafood, bovine meat, chicken meat, pork. The only other one that's greater is people exposed to other people in the community. So basically everybody eats. Everybody's getting exposed to antimicrobial resistant organisms through the food chain. And just one more way to look at this data, if you add these food sources up, they end up to be about 20% altogether. For this is only for one type of resistance in one kind of bacterium, in E. coli, okay? So we're 20% are attributed to these different food sources. And so you can see these people are getting resistant organisms from food. So that's kind of justifies the need to do something about this, okay? The other two things I mentioned, now I mentioned that those tables were one bacteria, one gene. We know there's an issue of co-resistance and cross-selection. So even if you're just feeding tetracycline, you may get resistance to something else. And this is just an example of a mobile genetic element. I picked this one out specifically that they found in a human caused disease, and you see multiple different resistance genes marked on the outside here. But what's particular about this, and it goes along with the theme of this particular conference, is if you do the analysis, these genes actually, this is a mosaic plasmid that has been collecting resistance genes. Basically, it's a One Health poster child for plasmids, okay? It's picking up resistance genes from various agriculture systems and collecting them, so we know they're being shared, okay? The other thing I'll mention is concentration of antimicrobials in animal feces. Because when an animal is treated, it doesn't metabolize or destroy all this. Much of the antimicrobial that's given, of course, it depends upon the specific antimicrobial, it depends upon the animal species. Much of that's excreted in an active phase still. So we have the issue of food contamination, we have the resistance of cross-resistance and co-resistance, and we have the issue of environmental contamination that we're dealing with, all because of subtherapeutic treatment of animals with antimicrobials. So, why don't people stop doing it? We know it's bad. Well, if you take away the AGPs under poor management conditions, you're going to increase your feed costs because feeding these drugs actually increases feed conversion. You have the potential for increasing mortality because you've got disease transmission, and you could have lot variability, say in chickens, where you've got some big ones and some small ones that results in contamination and slaughter and problems. But you don't have the antibiotic cost. I mean, and this is what they look at. And if you look at the numbers, and I pulled out some references here, how much loss per animal, and these are recent numbers, is it cost if they remove antimicrobial growth promotion? And you see for pigs, it's a little bit more, depending upon the situation, in between $4.50 to about $1 per pig. Chickens, it's a much lower level, like from a penny to a quarter per animal, okay? So the producers are going to lose money. However, what we don't take into consideration in this is that there's other individual and societal benefits. Okay, so the producer loses $5 a pig, but the Public Health Agency doesn't have to pay for those 5 million, well, actually not all the antimicrobial resistant infections are from food, and we don't know exactly how many of them are. Maybe we say 20%, but we don't have to pay for those extended hospital stays because the organism is resistant. We don't have to pay for the increased mortality. So there's a balance here that we need to take into consideration at the societal level for the benefit for the producer versus the benefit to the society. So this continues to be done. Now, I know we're running behind, but being a professor for 20 years, this is the 7th PowerPoint presentation of the day. We just have a little musical interlude here, okay? Can I have someone come up here and volunteer to sing "O Canada"? No volunteers.
Joe.
No, hey, let's look here. Why did no one volunteer? Right? Maybe you weren't aware that I was going to ask you that. You were unprepared. You had no idea. You didn't have any intention to know that I was going to do this. Maybe this is it. I mean, even if you weren't aware of it, you're not motivated to come up here and sing, right? Would it be more motivating to tell me that you had to pay me $100 more to come up here and sing? I would get fewer volunteers, right? I know you Koreans like karaoke, right? But I don't know what your skill level is. I know you don't want me to sing up here, okay? Because I don't have the skill to do that, right? And then let's see. Do you even know the words? Not in French, maybe just in English. We've got bilingual anthem there, right? And then the other thing is, this is a beautiful venue here, but maybe it's not the appropriate place. You know, we don't have any backup music, we don't have the lights, you know, we don't have the Canadian flag waving and all that kind of stuff. So maybe the environment wasn't conducive to you participating in this little exercise here, okay? Again, the acoustics of the room, who knows? I guess you don't get "O Canada" song today, right? But the idea is, I mean, I could have told you beforehand that I was going to ask someone, but awareness isn't really enough to change people's behavior. You didn't volunteer to come up here. Awareness, in some theories of communication, account for only 5% of people's behavior change. We have to be motivated, okay? Farmers that are going to lose money aren't necessarily motivated to adopt a behavior change. The other thing is the skill set. If they don't know what to do, how can they do it? I ask you to sing it, but you don't know the words, you don't know what to do. So how do we get, I mean, just to tell people, oh yeah, we stop AGP, stop AGP. Everybody knows that chorus, okay? That's the short one. But how do we get to be motivation? And then the last thing, they have to have the opportunity or an event to have that enabling environment to make it suitable to be able to adopt that behavior change, okay? And these would include things, training, resources, and enabling environment. So, put that in the context of AGP, all right? What are the context of AGP or stopping growth promotion that we need to consider? So we have economic, social, and legal and technical. We just heard one about legal, and you heard maybe a couple issues where we've got problems with adopting or lack of legislation, okay? So let's just look at these a little economically. I mentioned the cost difference. So FAO's got some publications we're coming out on the economic cost of foodborne AMR. Legally, Carmen's got work. We had some previous work done about how to review AMR relevant legislation. The social aspects, we've done a lot of knowledge, attitude, and practices surveys in different countries around the world, trying to understand why people are using antimicrobials the way they are. And then there's the social aspects as well. And then technical. And one question is, how do you stop, what do you do to replace growth promotion antimicrobials and still maintain the economic benefits by having the growth? So let's just look at these, and I'm going to go through these other ones rather quickly. You can look at the slides later. But, you know, economically, we know there's an economic cost to society, economic cost, a health cost, loss of productivity, decreased livelihoods, loss of trade, loss of product value, a number of things, okay? So we need to figure out that make it beneficial for farmers to adopt a change, okay? Social cost. Again, this kind of looks like this co-creation where you've got raising awareness and communication among multiple sectors, getting people to work together, okay, and motivate for behavior change, not only awareness raising. Technically, this list could go on and on a little bit more. The easiest thing to say is don't use them, okay? But then what do you put in their place? So we need to provide options and strategies for a sustainable, productive agriculture production, okay, in the absence of growth promotion, right? And part of this, and you've heard Alejandro talk about here, if we were going to change something, we need some surveillance systems to know whether are we going up, down, staying the same, what's going on, right? So that puts this in place. And then last, again, we could have legal strategies for growth promotion. We can have, adoption of laws or even strengthening here other laws, about environmental contamination so that the manure doesn't get into the waterways and get into the irrigation and get on the fresh fruits and vegetables. So there's a number of adequates. So in effect, what we're trying to do is put these practices together in a resource manual or technical manual so that users are interested, maybe we just call them stakeholders, right? Who are stakeholders? Whoever's involved is going to be participating and know what to do. And we've got a whole list, and this list of stakeholders could go on. Every one of you in this room is a stakeholder. Thanks for being here. But then who do we engage and how do we help them? So this slide just speaks a little bit to the alternatives. So we have a number of alternatives. So alternatives are available right now. And the issue with antimicrobial growth promotion is they worked really good when production practices were horrible and disease was rampant. But as we increase the health of animals and management and nutrition, that marginal gain by using growth promotion has decreased and decreased. And we can get it to a minimum and maybe even positive because I know in the United States they invest a little bit more. And one producer said it cost me 10% more to grow chickens without antimicrobials, and I sell them for 50% more. So in effect, he's actually making more money by not using growth promotion. So these are just some examples of different feed additives that are available or changes. So trying to, hopefully I caught us up a little bit on time. I'm happy to have questions, and I'll make a few comments at the end of the day and some of the take-home messages. But so I think I made it clear that this growth promotion is really a risk, not only to humans, animals. So now the treatment of bovine pneumonia can't work because we've generated resistance. And environmental contamination. Food safety is critical here, and because food safety is a key element of food security, without food safety, we don't have food security, okay? So it's not a simple matter of saying, okay, stop, go ahead, right? There are tools and approaches available, and that through the ACT project, now these are just simulated covers that we're working on. We'll have 2, phasing out antimicrobial growth promotion in animals, kind of outlining these social, legal, technical, and economic components of what has to be considered. And then also, we had a meeting, I think it was last month. What month is this now? It was in July. I guess we're already in September. About expert meeting on different nutritional and feeding practices which can be used to help productivity in the absence of antimicrobials. So I'll just close it with that right there.
Thank you, Jeffrey. In interest of time, I do not want to take any question at this moment, and I invite him again when we wrap up the session at the end of this presentation. Okay, we have 2 more presentations ahead. It's ACT project in Asia and also ACT project on the Latin America. So I'd like to invite two presentations. First speaker, Peter Flanagan, talk about the Asian Act project, and followed by Maria Gatica and Latin American Act project. So, okay, let's start with Peter Flanagan, the ACT project in Asia. Please join me. Welcome, Peter Flanagan, for his presentation.
Yep. Can you hear me?
Very well. Yes.
And can you see my slides?
Oh, not yet.
It says screen share.
Okay. Yep.
You can see the slides?
Yep.
Okay,
Yes,
I'll
yes.
start.
Yep.
Okay, very good. So, hello everyone. My name's Peter Flanagan. I'm the regional project coordinator for the ACT project in Asia. And I know that Myung Sing Choi has already talked about the ACT project in general. So I'm going to give you a little bit more specific information about the activities of the ACT project that are going on in the Asian region. So first, I always like to start a presentation with good news. And the good news is I promise that I will not sing "O Canada." So I'll spare you that pain, okay? So the ACT project, and second thing I wanted to mention is that I'm actually contacting you from Pakistan, where we just had a regional meeting of our group in Asia. So if the power cuts out, it's because of the situation in Pakistan. So cross your fingers, I can get through the next 20 minutes or so. This is going to be just a brief introduction to the activities that have been going on under the ACT project in 4 different countries in Asia. And I like to show this slide with the flags for 2 reasons. I like flags, and the second thing is that each one of the flags for the 4 countries, Cambodia, Mongolia, Nepal, and Pakistan, strongly reflect the cultural differences between each of these countries. And in some ways, the cultural differences are making this project more challenging, more interesting, but it also presents a whole new set of issues that you might not see in a project where the countries are more uniform. And at first I questioned, why are we doing a pilot program in 4 different countries that are so different culturally, as far as their food is concerned, as far as the geography is concerned, as far as their farming traditions are concerned. And I've come to realize that actually as a pilot project, the ACT project in Asia is actually a perfect place to find out what can go wrong and what things are actually in common when it comes to trying to control AMR. So as I mentioned, all of these countries are rich, but they're rich in the variety of the geography, the environment, the conditions, farming methods, and food habits, and lastly, and very importantly, sort of the political situation in each of the countries. So the project is facing a wide diversity of challenges, and each of these countries have different priorities. So unfortunately, I cannot present this presentation in person, and so online it doesn't work quite the same. But when I showed this same slide the other day to a group of Pakistanis, they were very easily able to identify which countries are involved. And so I'll just show you the picture from a recent mission that we had in Mongolia, which is down in the lower right-hand corner. And you see sheep. And if I panned my camera another 90 degrees or even 360 degrees, you would see sheep, and then you'd see more sheep. And so in Mongolia, the project is focusing on this arid land and sheep production. If you go to the slide on the opposite side on the bottom, you'll see this is pig raising in Cambodia. And the challenges that pig raising or swine raising there are completely different from what we saw in Mongolia. And certainly for cultural reasons, it's not anything that we are even concerned about in Pakistan, because pigs just are not part of the cultural tradition and diets. So the group the other day in Pakistan was able to identify exactly which country we were talking about depending on what photograph I pointed at. So this makes the program exciting, but also challenging, as I said. So I'm calling now from Pakistan, Islamabad, but normally I'm based in Bangkok, in Thailand, at the FAO RAP headquarters. And fortunately, over the past 18 months or so, the AMR group has actually grown, where we now have 8 AMR projects based at RAP. And so there are many projects that you've heard before, like Fleming Fund. The EU has activities there. USAID actually has 2 projects. FAO itself has 2 AMR projects, and the MPTF has a couple of projects. So fortunately, we have been able to build a team. I lost count of the number right now, but it's probably around 10 people who are actively working on AMR projects based out of the regional headquarters. And this has been great because we've been able to share some of our experiences for sure. And we actually have been able to piggyback or work together on various projects. So for example, in November, we're planning to have an ATLAS mission to Cambodia. ATLAS is the laboratory assessment. And we are sharing the responsibilities of our project with the Fleming Fund, where there's some overlap. And instead of having competitive projects, we're actually finding ways to cooperate and assigning responsibilities between projects so that we actually make the most of the funds that we have available. So this is the good news. So I know that Min Singh has mentioned a couple of the activities that the ACT project is engaging in. And so even though I introduced this topic with saying that there are lots of diversity, there's actually a lot of activities that we're trying to do in each of the countries. And so the PMP assessment, or the Progressive Management Pathway assessment tool, is something that we have applied in each of the 4 countries already. And basically, this gives us a status of the AMR activities in each country, and also it's a chance for us to see where the National Action Plan overlaps with the Global Action Plan on AMR. Second thing that we've done in most of the countries is we've done ATLAS assessments to look at the laboratories, to see what the laboratories' capacity is as far as testing for AMR. And you can imagine that there's quite a bit of variety in the laboratories, central laboratories, regional laboratories, and their ability to actually perform the basic AMR testing, but also and how often they actually do it. So there's the quality and there's a quantity issue that we have to address, and it varies quite a bit between countries. Finally, a third thing is, I think Alejandro has already talked about the InFarm program, and we've started to introduce InFarm on a pilot basis in a couple of the countries, and certainly it's a hot topic for us as far as our work plan. So actually, the reason that I'm here in Pakistan now as we're working on our work plan for 2024, and in-farm actually figures largely in our activities. And finally, we have a tool that we're calling the ACT tool. And this tool is actually looking at the legal status of different countries, see where this legislation exists, see where the gaps are between where we think we want to be and where we actually are. And this project focuses highly on Codex standards. And so we're looking to see in particular where Codex standards have been adopted or not adopted, where they've been integrated into regulations or even legislation. And there's also a technical component to this. So we've actually been in each of the 4 countries, we've conducted the ACT tool, and the legal assessments are underway. So this slide briefly summarizes what we've actually done. So in all 4 countries, we've conducted the PMP, or the Progressive Management Pathway Assessment, and there are actually 4 levels of assessment. So for people who are not familiar with this management program, it's based on other similar management programs that are conducted by FAO. So maybe some of you are familiar with the FMD PMP. So this is similar, except that it focuses on AMR. And in the 4 countries in Asia, the consensus is that basically everyone's at the beginning. They've done some activities, but they have been quite limited, or limited either in the size of them or the duration. So one of the things that we have found is that when there are AMR projects, that's good. People will our countries will do testing. They'll take the samples to the labs, but once the project money has dried up, then the testing stops. So there hasn't been any sense of sustainability or growth or commitment to moving on with the testing in the laboratories on a national basis. And you can understand that, at least I can understand it, when you realize that AMR is not the most glamorous topic. And I've mentioned this several times. I'm a little jealous of my colleagues who work on FMD or lumpy skin disease, because the beauty of those terrible diseases is that you can see them. So if a farmer has a FMD or lumpy skin disease, it's easy for them to get excited and complain and make a push. AMR, unfortunately, is not one of those things. So yesterday we visited several farms in Pakistan, and you talk about AMR and they don't get it. The farmers don't see see what AMR looks like. All they know is, whether their animals are growing better or not, whether they give the animals antibiotics or not. So that's one of our challenges that we found, and we have to come up with ways to make it more obvious for governments and farmers to see what AMR is. So one of the ways that we can do that indirectly is to actually generate data. And so this is where the Atlas comes in, and I know that Alejandro has talked about Atlas already, so I won't belabor the point, but we're conducting Atlas missions in each of the 4 countries, and 2 are complete, and we have, well, actually, 1 is complete, 1 is happening next week in Nepal, and then Pakistan and Cambodia, we actually held off on doing an Atlas assessment because laboratories have been assessed about 4 or 5 years ago under Fleming Fund. So we will plan to do these Atlas visits in the next couple of months, actually, and as follow-ups rather than as getting the baseline. So, but the Atlas missions are really important for us because if we can generate data from the laboratories, then that gives us a chance to show to government officials, like, this is what AMR is, this is what the actual status is, and we can use the data as evidence. You know, it's not as nice as a lumpy skin disease or lesions between the claws of a cow's foot, but it's at least someplace that we can show some evidence. And finally, the ACT tool is a legal assessment. And as I mentioned earlier, the ACT tool is already been applied, where we use local lawyers to discuss this tool, ask a series of about 150 questions, where we're asking, you know, what is the legislation? Does the government have any kind of policy on growth promotion, on pharmaceutical use, on legislation on imports and exports of medicines, and are prescriptions required? And questions like that, very practical questions that say, okay, what are the regulations? What's happening? So the preliminary summary for all 4 countries actually is, a lot of the countries have a lot of regulations or some regulations, but the next step is, are those regulations enforced? And this turns out to be almost universally true in the 4 Asian countries. The regulations are on paper, but no one really enforces them. So yesterday we were in a veterinary pharmacy, and we easily could have bought all the antibiotics that we wanted, or all the antimicrobials. And it's a challenge because we know that it's there, but there's just no kind of enforcement. So that's a very general summary about what is going on. The laboratories have very limited and no sustainable capacity. The action plans will do some activities, but they're not really very well developed, not very strongly supported by the countries. And the legislation is in place to some extent, but it's not enforced. So as Min Sing mentioned, the project has 3 outputs. One is to increase awareness. Second one is to expand surveillance. And the third one is to support the government to adopt Codex practices. And along with that is adopting good practices and encouraging farmers to start to use more and more biosecurity measures to ensure that their animals and their products are healthier, and that they can produce with less antibiotics or antimicrobials in place. So you can see some of the situations. The farm that you see on this picture is actually a Pakistani farm. It's actually very nicely arranged. I wish I had the photo from the farm that we visited yesterday, because I think all of you would become vegetarians if you saw the conditions that the animals were raised under. And there was abundant use of antimicrobials, not because the farmer was aware that there were antimicrobials in some of the feed supplements that they gave, but they just know that their animals seem to perform better when there are antimicrobials in the feed. So I think the point is that when it comes to raising awareness, farmers don't necessarily use the term awareness as far as AMR is concerned. What their awareness is, does the animal do better on this feed as opposed to, or on this supplement as opposed to not having this supplement? So I think one of the lessons that we learn when we visit rural farms is, the farmers understand. We just have to learn how to speak their language. And I was at a fancy FAO meeting the other day, and I looked at all the people wearing suits, and they were talking about how we have to learn to communicate with the farmers so that they can get the message. And one of the things that we realized is that we were just talking to each other. So one of the things that we want to do in the ACT project is to make sure that we get out into the field, out into the farms, and actually talk to the farmers and hear what they say. And it's our responsibility to interpret what they mean when they talk about growth promotion. So that's the first message. But what you can see in this lower right-hand picture is actually a situation in Pakistan where we have people who are actually training locals on AMR. This was part of the World Food Safety Day efforts, and we're just trying to get the word out about as much as it's important for us to get the word out, it's important for us to hear what the farmers are actually saying so that we can understand what they understand, what their understanding of what AMR or what growth promotion is. So, like I mentioned earlier, the second thing that we're trying to do is to expand surveillance. Actually, we've been meeting for the last 3 days with our colleagues in Pakistan, and what we're trying to do now at this stage, now that we've done our tool development and our assessments, is actually to develop capacity. And so capacity development for us means developing little programs, pilot studies, and collecting samples from various sources and testing them. And like I said earlier, this is our opportunity for us to generate some data that just doesn't exist and provide that to the government officials so that they can understand where we can give evidence, basically, show them that we have, that there's AMR or not in certain sectors in their communities. So to do this, we've actually reviewing several pilot programs. There's one that's actually ongoing now in Mongolia that's looking at slaughterhouse samples from, it's actually from cattle, but it's getting started. It's happening. We're collecting data. And I'm expecting that beginning of next year, we're going to have in Pakistan samples that are collected from aquaculture and poultry, and in Cambodia, we'll have samples that are collected from a poultry industry in a selected area. Similarly, we'll see something, we're actually discussing 4 different projects in Nepal. One to look at growth promoters that might be in the feed that's fed to the animals, also an aquaculture program, and also a poultry program. So lots of things that are getting developed, notes are developed, and plans are being developed that really expect that at the end of this year, we will start getting more and more projects and more data collected. So finally, along the same lines, we're trying to improve laboratories' ability to actually conduct tests. It's one thing to conduct tests, it's another thing to conduct tests that actually have meaningful results. And so several of the labs that we have looked at, they can do tests, they can test for AMR, but we're not sure about the quality of the results. And so the ACT project in Asia has been supporting the proficiency testing programs that are actually offered through the Chulalongkorn University, which is the regional reference laboratory based in Thailand. But next week, we actually will have representative experts from Chula who will be going to Nepal to assist with the laboratory training there and with the goal of improving the quality of the testing that goes on. And finally, we're supporting legislation. We've conducted our ACT tool, and we see where the gaps are. And because there are gaps in many different areas, the ACT project itself in Asia has decided that really the place that we want to focus on is is working on prohibition on growth promoters. And there's been some resistance, partly because people don't want to change the habits. And there's also been some resistance, or a lot of resistance, or negligence maybe is a better word, because people just don't know what we're talking about when we talk about growth promotion. So it's incumbent upon us as part of the ACT program to increase awareness, use the numbers that we generate from surveillance to show the government officials the importance of adopting Codex standards. And going back to the very first slide, promoting growth, policies or Codex standards is going to be a little bit different for every country. So this is one of the challenges, I suppose, for the 4 countries, is that how each country will have different standards that they will want to adopt. And our challenge is to identify the standards that the countries are willing and able to adopt. So this will be a long-term project, I think, to get people convinced that this is an important thing to do.
Thank you. Okay. Peter, may I interrupt you? I'll give you 1 more minute.
Okay,
because we
very
are
good.
okay.
Okay, very good. I'll go very fast. So this slide just summarizes some of the activities that we've been doing. We have a number of global initiatives. We have a lot of our activities are more focused on the country level, and we meet with veterinaries. We go to the veterinary schools, veterinarians, herders, and so on. And then we have a lot of general communications through websites, traditional media, and social media. So I think I've covered a lot of this information on the slides already, so I won't go into the detail in the interest of time. But just to say that we are very active in all 4 countries, working with the government, working with farmers, and trying to do what we can to reduce the use of antimicrobials at the feed level and the food level. So, summary, once again, we're working with a number of different organizations in a cooperative way so that the money that we get from the Republic of Korea is actually, what's the word they use now? Synergized, built. And so we take a little bit and we make it stronger by magnifying it and cooperating with different projects to make sure that we don't overlap, don't compete, and work in the same direction. So that's the end of my presentation. I thank you for your attention, and I think there's a little question and answer period after Angela speaks, so I'll stay online.
Okay, thank you very much. You're sharing the good information and experience. And we really appreciate your efforts. You're doing action in an active project in 4 different Asian countries. I want to hold you until another presentation in the Latin American Active Project, and then we will close after the presentation, and then we'll ask if we have, we will ask you a question. Okay, thank you very much, Peter. Okay, last presentation will be that the active project in Latin America is presented by Maria Katika as FAO project coordinator. Please.
Thank you, and greetings from the other side of the world. Just 1 minute to share.
Okay, before you start, Maria, could you finish up in 15 minutes, if you can?
Yeah, I will try. I will try to go fast.
Thank you very much.
Thank you. So let me know when you see the screen.
Yep.
Is that okay?
Yep.
Okay. So thank you for this kind invitation to participate on this conference. On this occasion, I will talk about the work of the ACT project in Latin America and the Caribbean. So this presentation will focus on a little bit of history of the work that FAO office in the region has been doing on AMR, our first findings from previous work on AMR in the region, what are we doing in Colombia and in Bolivia, and some of the lessons learned from the project so far. So the FAO regional office begins its work on AMR in 2016. At that time, countries were responding to the call of the Global Action Plan to develop and implement the national action plans for AMR, including agriculture. And actually, that was the first project of the FAO regional office focused on. And one of the first steps, as you know, for the implementation of any national action plans is for the health authorities to have information detailing the situation on AMR, so they can be able to design and to implement and to manage appropriate strategies for each of the countries. So at that time, the team began to explore information regarding AMR, and they found that there was a big gap on information in the agriculture sector. So to that date, so far, the only two approximations that were available were the work that the WOHA was doing on the annual reports regarding the antimicrobial use in animal production, and the work that the TRACS was doing regarding the monitoring the countries and the implementation of the national action plans, but there was little information regarding agriculture. So that is why the FAO regional office in Latin America began to work and to provide countries with specific information regarding AMR data from the food and agriculture sectors. And this was a first approach on general aspects of AMR, taking into account the risks of AMR from the food and agriculture sector, but also considering some mitigation measures that were available in the countries that could be regulations, private or public programs, or anything else related to the implementation of good practices. And all of this without considering any specific characteristic of any particular agent. So since 2016, FAO has worked in 5 AMR projects in the region, and we have been able to cover 15 out of the 33 countries in the region. So from our first findings in the project, we saw several challenges to address related AMR risks. And just to mention a few, for example, related to farming practices, we identified that, for example, there was very little use of the registration of animal population in the different farms. We also saw that there was a big lack of implementation of biosecurity measures. We saw the absence of a veterinarian for the diagnosis of animal diseases and infections. We also saw that the lack of implementation of welfare measures. And regarding antimicrobial use practices and medicated and non-medicated feed, for example, we saw that producers were using antimicrobials classified as critically important, or also the use of antibiotics as growth promotions, or even we saw some problems with the compliance with withdrawal periods. We saw also that there was a lack of surveillance programs related to foodborne diseases and residues from antimicrobials. And one other thing that I think it remains really important is environmental practices, specifically how the producers and animal farmers disposed the organic and chemical waste into the environment. So besides these risks in practices, we also saw what we called at the moment some gaps in these sustainable factors. This is awareness, risk communication, training, and of course, AMR surveillance and governance. That many of them are the topics that we are talking about during this conference, and we are also incorporating in the ACT project. So with this slide, just to show you or to make visible in a timeline the AMR project that the FAO office here in the region is working on. So as I mentioned, the office started working on AMR in 2016. And just to say that from that point, it is important to mention that every result from each of these projects has served as a guide for the development of the next projects on AMR. And this is an approach that has allowed us to build and organize in a more sustainable work, in a more sustainable form, the work on AMR for the region. So this is just for you to know a little bit more about Bolivia and Colombia. Both countries have a very similar area, so they have 1.1 kilometers squares. In both countries, the most important productions in terms of animal populations are poultry and bovine. And just to put in a few examples of the work in Bolivia and Colombia regarding AMR, both countries have participated in FAO projects before, and both of them have advanced in the development and application of national action plans. Colombia approved its national action plan in 2018, and Colombia is working on getting this approval. And also both countries are working or have an intersectoral committee for AMR, and they are working on its formalization. So in 2021, we were presented with the challenge of putting together a work plan to support the implementation of codex standards related to AMR in the region. And we assume this challenge considering the 5 objectives of the FAO action plan, and of course the products and the outputs of the ACT project, but also the experience that we have from previous projects in AMR in the region. So the ACT project, I think it can be divided in 3 stages. The first stage is the situation analysis. The second one is the implementation of actions, where that is the point that we are now. And a third stage that is rather transversal and refers to the follow-up of each of the project activities and the commitments of the countries during the project. So during the first stage of situation analysis between 2022 and 2023, we apply several tools of AMR, some AMR tools from FAO, that is the PMP and Atlas that Peter was also mentioning. And these tools allow us to know and to recognize the process and to prioritize the needs in both participating countries in the region. And it was these activities that the ones that allow us to create and to form the work plans for each of the countries. So for the implementation of activities, it has been very important for us to agree these activities with our national counterparts, this to know the context that we will be working on and to give them the sense of ownership of each of the activities that we will be applying. So regarding the follow-up, it is also important to mention that during the situation analysis, we saw many activities that we could do, but we selected some of them to be in the project because of the scope of the project. But we took the commitments to follow up every activity in the project, but also the activities that the country committed to do by their work. So, as I mentioned before, we had this situation analysis that give us that that allow us to see how the countries were and where were the most important gaps. So, for example, in terms of governance, we saw that there was great interest in addressing AMR in every sector. And the project and also the countries were able to identify which were the key stakeholders. For many of them, these first workshops were the first opportunity for them to meet and to know the activities that each of the institutions were doing. We also saw in governance that there were some difficulties in the formalization of the multi-sectoral committees, probably because there is a need to build more governance activities and initiatives. We were also able to see that there was a need in reviewing the legal frameworks. That is something that we are doing, as Carmen has mentioned before. And regarding awareness, we saw that the countries highlighted the need to evaluate the awareness level on AMR between the different stakeholders. Regarding good practices and responsible use, we saw that the countries have programs related to best practices but there were only a few of them that included AMR or antimicrobial use. So there is also a need to reinforce the training on AMR and best practices in the countries. And regarding surveillance, countries have moved forward in the implementation and development of different surveillance programs, but there is room to implement improvements. We also saw some difference in the capacities between the laboratories within the countries. And we also saw that there is important data collection on AMR, but this is not systematically used. So we see this as an opportunity to support the countries on building capacity on epidemiological analysis, and of course on how to use this information in communications and how the authorities can use this information for decision making. So what have we been doing in the project in this last 2 years? So for output 1, related to increased recognition and the use of codex standards, at a regional level, along with CCLAC, we started a webinar series on codex standards for Latin America and the Caribbean. And to date, we had 4 modules with different topics related to codex. And we are hoping to maintain these activities until 2025. And as at a country level, the project has worked on communications and awareness products tailored to the needs of each of the countries. For example, we have been doing some recognition of AMR heroes, the development of videos from national influencers regarding agriculture. We have had some high-level meetings, ludic experiences to raise awareness between different stakeholders, among many other activities. And at this moment, we are also working on the preparation of radio messages that will allow us to deliver the message of AMR Foodborne to the different stakeholders and a very important group for us that are farmers and veterinarians across the country. So in Bolivia, related to the output 2 on AMU and AMR surveillance, we are working, or we will be soon working on laboratory capacity building, and specifically on reviewing laboratory protocols for AMR susceptibility testing, but also quality assurance to make sure that the data they are collecting are in good shape for the surveillance systems. And the country is also working on development AMR surveillance plan in primary production that we hope to expand in the next few years. And of course, in-farm is an important activity in our project for next year. And regarding the adoption of the Codex standards, the country is preparing the prioritization of food and agriculture sectors. They are also working on creating an agriculture committee for AMR that will help them to discuss some technical issues on AMR, but also to reinforce governance from the food and agriculture sector. They're also working on the identification and adaptation and application of manuals and trainings. As I mentioned before, they have many manuals, but they need to be updated and to include some AMR messages. And also they are working, of course, in the strengthening of the regulatory framework. At the moment, as Carmen mentioned, we are working on the development or the finalization of this report regarding the implementation of the Act tool that will reveal the most important needs for the countries regarding regulatory measures. And of course, we are working with the country to set the new priorities or the next priorities to develop the work plan for the next years. So in Colombia, on the other hand, for output 2, Colombia is working on the design of a surveillance plan for AMR with the help of IICA. And after that, we will take the activity of strengthening the intersectoral work on AMR surveillance. So we make sure that this plan that is being created during this year is correctly applied or can be applied. And we are also working under the project in implementation of an epidemiological unit in the agriculture sector. So this will help the country to gather all the information that they have, to process this information, and to use this information. And of course, in-farm is also an activity under output 2. And under output, 3, regarding the adoption of Codex standards, the country is working on the antimicrobial use characterization, and also in characterizing awareness between the different between farmers and veterinarians. So this will be a great help for us to include key messages in the manuals and trainings that the country is identifying at this moment. And of course, too, the country is working on these legal frameworks for AMR and the application of Codex standards. So to finish, I just wanted to share with you some lessons learned from the project to this point. At a regional level, the implementation of the project to date has shown us the importance of basing the activities on previous experiences in the region and the countries. This has allowed us to take a step-by-step construction of the different actions related to AMR. And specifically through this regional webinar for Codex Standard, we have been able to see the great interest of the region and different countries within it about Codex standards. And at a regional national level, we have seen that both countries have excellent professionals. Both countries have shown great interest in AMR, but also the project. And this project has provided support in one of the many concerns that any country might have. And one important thing is also that the project has shown the countries the progress and gaps through concrete methodologies. So in the future, if they decided to apply these methodologies and tools again, they will have comparable results. The project has also helped in encouraging the work, the intersectoral work. As I mentioned, in many occasions, it is difficult for the institutions to learn about the work that others are doing and how to identify mutual points of work. So the ACT project, it's been really active on that. And it's also important to mention that the progress has been made prioritizing actions according to the country's own needs and resources. And the countries and country counterparts have been involved on every step of the project, so they can make it their own. And I will also like to highlight that the project is working to deploy activities that provide some tools for sustainable work in the countries, because we have seen that in many projects, after they are done, there is the countries feel that absence. And finally, we should always take into consideration the socio-political context of the countries. And this forces us always to be flexible and on how to face these possible problems that we may encounter during the project implementation. So I hope to have made this presentation on time. So thank you. If you have any questions, I'm here to answer.
Okay, thank you very much, Maria. Peter, are you still there? Peter?
Yeah, I'm here.
Okay. I'd like to ask any question or comment to ACT project from the floor. Really appreciate, before I ask the question to the audience, I really appreciate the two project coordinators and the FAO's efforts to do things happen in Latin America and then also in Asian countries. So through your efforts, we believe in the future AMR awareness and capacity building can help to implementation called practice and the guideline of integrated the AMR surveillance and monitoring programs and at that places. Any comment, suggestions? If you don't have, then thank you again for your efforts and your experience sharing with us. And please keep going, and we can assist you, anything we can do. Thank you very much, Peter and Maria.
Thank you.
Okay, thanks.
Okay, next 5-10 minutes, we will wrap up all the session. and all this symposium, I would like to invite Jeffrey Legion to present 5-10 minutes to what we have learned through this symposium and what we can do in the future symposium. Then we can have an official closing.
Thank you very much for that time. I'll try to keep my message brief. And actually, I'm going to skip the first couple of slides and cut right to the chase. I know it's late in the day. I'm going to give a demonstration. You've had so many PowerPoints. Need a volunteer. Marcus, I see your hand up. Come on up. Thank you, everyone. You can bring your phone if you want. You might need it. Yeah. Just, oh, well, he's doing that. Let me just put this stuff out.
Thank you.
This is not for you. No, just leave that there. You can stand right here.
You're gonna make me work now?
No, okay, don't worry. If you can hold this, please. All right. My goal is to get that adequate amount of maybe not so nutritious but safe food. Right? Your goal is to stop me. All right. When you do that, you can try to push me off the edge there, over here. I'll try to go that way, right? And when I fall, maybe I might be injured. I might feel bad for a little while, right? And maybe I'll break a leg, end up in a wheelchair for the rest of my life, have a long-term disability, or I could die.
Yeah. That's the risk.
Could happen to you, too. All right?
Okay. Got it.
So I need a countdown. 1, 2, 3, okay, please?
1, 2, 3.
Oh, wait. I got to give you the strategy first. Okay, so I know Marcus is a few days older than me. So I'm going to give him the advantage. I'll put my hand behind my back.
Thank you.
Right? Or actually, better yet, I'm going to hop on one foot. All right?
Okay, that's better.
This is better, right?
Yeah.
So I can go there.
Okay.
But I'm a little wobbly here. So I need an approach. And like any approach, any sport, there's the offense and the defense. So I'm going to try to build my offense and take away his defense. So, first of all, you don't need any nourishment. Let me have that back anyway. Try to make the environment poor for you. Do you have your phone? You said you brought it up.
My phone also.
No, you don't need it. I just don't want you to exchange any information with any of your friends on how I'm going to get over to attack and get that food, okay?
Right.
All right? Oh, wait, one thing. The other thing I can do is if I, just let me go by over here.
Nice
No, just a second. I'm going to pick this up. And if you look on page 20 of this conference, there's a bibliography here is about Marcus. It outlines all his strengths and weaknesses, well, at least his strengths. I know some of his weaknesses, so I'm going to keep this with me. Okay. And if I go over here, you've seen this before too, right? This is the Compendium Act. So I'm going to take these things. So with these things, it kind of counterbalances my handicap here, so I can walk on 2 feet. All right?
Okay.
So I'm going to go. But wait a second. I want some help. Jorge, please. WHO. Natasha, come up here, please. My partners, quadripartite partners. All right. All right. Let's see. Sang Mok. Yes, please. Anyone? Kim. Where did she go? She was here a second ago. Yeah, okay. So we got all these people here. Right?
Okay.
Now we'll try this again.
Not fair.
I'm trying to shift the balance here. All right, you're on this side. No, not for him. Oh, I forgot one thing. So there's a couple things. Another thing. So if you don't know, he's my supervisor. He didn't know he was coming up here. So I could put myself in financial my livelihood could be at stake here. Okay.
You're committed, it's nothing else.
So that's another thing that I forgot to mention that we had a problem with here. So, you guys can call anyone you want too, I suppose. I guess we can't mess this, we don't want blood in here, so maybe we'll just call it quits for here. I think whose side are you going to win? Who votes for our side over here? Raise your hand. Why don't you come up too? That would be good. Oh, yeah. I would really like to have Dr. Park with his experience and expertise.
I'm in charge.
And I'll be done here in a second. And we also need geographic representation and young minds with innovative ideas. Oh. And we could take on this guy, which, you know, on all the stories you have to change the names so the innocent. We'll just call him Sam instead of Marcus, okay? We'll call him Sam. So we'll stop there. You know who's going to win this game,
yes.
All right? Thank you all for your presentation. And then I have like 4 slides I want to show you, okay? You can take that, I suppose. Okay, thanks for playing along in that game. As I mentioned, I love coming to Korea. I love the culture, the food. I haven't done the karaoke yet, but the culture and interesting things going on. But just to summarize what I've just outlined here, okay? So our goal here is zero hunger. I can spy it right there. And we want to make sure if we have from food security, adequate food, nutritious food, that doesn't really cut it. But safe food. We want safe food. And that food can be a variety of sources, okay? We've got fats and oils, proteins, animal origin food, vegetables, things like that, whole grains. This is the year of the millet. So if we're here, okay, and we want to move up there to get that safe food, seems like an easy path. But the issue is, and if this works right, we've got these obstacles, these barriers, these threats. Can't be. Salmonella, right? VREs, these escape pathogens that Dr. Park mentioned, MRSA, ESBLs. We've got all these things that we need to get to. And so what we do is if we have these ways to power ourselves up, grab the book, inform ourselves, get more information, right? Then we can superpower ourselves and eventually take that and move it forward, okay? This pushes those pathogens out of the way and allows us to move to have that safe food supply, food security, right? Does this framework look familiar to anyone? Anyone Korean? Somebody's smiling, no? Even the marks on the ground? If I get over there, I've won, okay? I have won. But if you look at this framework, anyone know what this game is?
Squid Game.
Squid Game. That's a children's game, right? This is a very popular movie. That's for fun. This is a fictional story where people are dying. This in the middle is real. Foodborne illnesses are killing people, okay? Antimicrobial resistance is a problem. We need to get through there. We can power ourselves up with the information, the data, the implementing acts, and coming together. The 2 things that if you want to take home from this message, we need to empower and engage. So empower each other to know what to do, how to do it, when to do it, why to do it, and then help other people to do it. Engage with others. Empower and engage. And in fact, AMR is not a game like the Squid Games. It's not a make-believe story. Lives are lost, livelihoods are being destroyed, and economic growth is impaired. So we all need to leave from this conference with this in mind and decide what we need to do. I told you if I got over there, I won, okay? I've won. Well, in effect, no game, no game. But the bottom line, if we work together, this whole team, One Health means health has won, okay? That's where we need to be, and I appreciate you all coming to this conference. I really appreciate being invited to this conference. The dialogue has been great, and I hope we continue to move it ahead. We're excited about what we're doing in the ACT project. We're excited about our collaborations with other people, and we just look forward to these continued discussions, dialogue, and engaging with other people to tackle foodborne AMR. So with that, I finish. Thank you.
Thank you very much, Dr. Lejeune, for your excellent performance. It's great, the message to all of us, what we can do in the future and the next generation through the overcoming the AMR issues. Okay, everybody looks a little bit tired, so now time to close the two-day symposium on AMR. I really appreciate the Korean government to be funded to implement this kind of symposium, the 3 consecutive years, and of 2 more years, hopefully, then we can do better funding to expand further activities. I invite the MFDS to close this all the session, please. Thank you very much, all the audience.
네, 제4세션을 이끌어
Yes, Professor Park, who chaired this session, all the presenters and contributors, a big round of applause for everyone. Thank you. So we have been here for many, many hours. So I would like to thank all the participants, both here and also online. And I thank all the presenters, the moderators. And so I would like to thank everyone on behalf of MFDS. Before you leave, please remember to fill out the feedback sheet and submit it before you leave. So, on that note, we will officially conclude the 3rd GCFA. I hope to see all of you in the very near future. Thank you.